We're really excited about our bridging scales in the ocean carbon cycle session today. I would first like to just say a few quick words about um, what our workshop's all about, what we're all about. I mean, this really sums it up, this slide right here, science, connection, community. That's what OCB is about, facilitating science, connection, and community. You can find all of our information for this workshop on the workshop website. The draft, it's not a draft anymore. The real agenda is right there. And you can also check out the gatherings that are happening. They've kind of increased over the last 24 hours. All of the satellite gatherings going on around the US and hopefully beyond. If you want to set one up, we really encourage you to do that. We had a lovely walk in Woods Hole yesterday, and I would love to see more of these happening. And if you want to post about them on Blue Sky, you can find us there, hashtag OCB 2025. So OCB is really it's a science networking, science community that's really sitting at the nexus of climate, biogeochemistry, and marine life. These green icons you see at the top here are really, they represent the scientific breadth of OCB. It's a pretty broad program, as you can tell from these icons, but we've really not only try to build and push science forward within each of these icons, but also across each of these icons. And that's really what the summer workshop is all about. We do this annual flagship summer workshop activity, sometimes in person and sometimes virtually, but they're really about cross-fertilization across these big green topic areas. And a lot of great things come out of them. Great ideas, proposals, collaborations, new workshops, workshop proposals, and new OCB research areas that get spun up because of the chats that happen. People are doing napkin sessions, napkin sketches over lunch, or maybe they're talking over Zoom and then setting up a time to talk more after the workshop. It's really about connection. All of the information about OCB is on the bottom half of this slide. You can find our website or subscribe to our e-newsletter, email us at any time, and follow us on social media. Blue Sky, YouTube is where we post all of the recordings from pretty much all of the activities that we support. The OCB program and project office are funded by NSF and NASA. And you should especially thank those program managers right now who are working really hard to ensure that OCB continues in some capacity as we move forward. So we're working really hard with them, and they are fantastic champions for this program and this community. I also want to thank Woods Hole Oceanographic Institution, which has hosted OCB since our inception in 2006. And that is all I have to say. I would like to introduce Zach Erickson, who is the lead organizer of the Bridging Scales session, and I will let him give the introduction.
Hi, everyone, and thank you so much, Heather, for that welcome to OCB today. Day 2 of the conference and the first day of the plenary sessions. It's really amazing to see everyone online, and just so happy that this community is still meeting virtually. So this session that we're going to start off with is called Bridging Scales in the Ocean Carbon Cycle. It's there's a number of organizers that are here, and I'm going to be giving a little bit of an intro talk to start off the session, and then a bunch of us are going to be doing, moderating and kind of leading a discussion at the very end of today. And then we have 6 wonderful speakers. I'll talk just briefly about what they're going to be presenting on at the very end of this introduction, but I'm trying not to steal any of the limelight from them. And then, of course, we have 11 wonderful lightning talk presenters as well. There we go. So when we think about the ocean carbon cycle, there's any number of different, kind of overview schematics we can choose. And I picked this one. This is one from Tim DeVries a couple of years ago, and it kind of shows the ocean, or really the carbon cycle in kind of its biggest form. And one of the things I like about looking at, somewhat simplified pictures like this is that you can really see the numbers that we're talking about here when we think about the carbon cycle. And in particular, one thing that a lot of us think a lot about and worry about is the anthropogenic signal, this 10 gigatons of carbon that's coming up into the air from humanity. And just the fact that that number is kind of dwarfed by the other fluxes in the system. You think about the 10 gigatons of carbon coming up into the atmosphere and compare that to the 90 or so gigatons of carbon that are going across the air-sea interface every year when you integrate globally. And it really just puts into perspective the kind of enormous role that the ocean, as well as the land, has in thinking about the broad scale carbon cycle of this planet. So on average, we think of the ocean as taking up something like 30% of the anthropogenic carbon. But as I said before, this is the product of a relatively small imbalance in the really large amount of carbon that's going into the ocean through the air-sea interface and going out of the ocean. It's this very small number. And when you look regionally, you can have much bigger imbalances, of course, but on the global scale, it's relatively small. And so if you change this imbalance just a little bit, you can really change the carbon cycle a lot. A lot of the carbon in the ocean is stored as dissolved inorganic carbon. You can see the number written down here, something like 37,000 gigatons of carbon. And, again, in this session, we're going to be talking a little bit about MCDR and the various strategies that we have to do marine carbon dioxide removal. And some of those strategies seek to just change this number just a little bit. And just a small fraction of a change in this number of dissolved inorganic carbon can dramatically change the whole system. And then the one that a lot of us think about a lot is kind of this biomass number, something like 5 gigatons of carbon. It's relatively small, perhaps, as a pool, but this does a lot. This 5 gigatons of carbon does a lot. And that's because there's an arrow that's not really on this plot, and that is because this particular figure is really thinking about just kind of the very macro scales of the carbon cycle. But there's this kind of arrow here that I'm labeling deep sequestration. And the biological carbon pump through this biomass flux allows for this kind of downward flux of carbon into the deep ocean, sequestering carbon along time scales. And it's kind of hard to put a number to this. And one of the reasons why is we have a lot of questions. How deep is deep enough? How sequestered is sequestered enough? What time scale does all of this need to act on? Kind of at the broadest sense, you think when you go deeper, you sequester for longer, but it varies regionally. And it's really important to think about these questions. And a lot of our presenters are going to be talking about, these questions in general. How deep is deep enough? How sequestered is sequestered enough? And this comes up a lot when we think about MCDR, but it comes up every time we think about the ocean carbon cycle, and especially the role of the biological pump. So thinking about the biological pump a little bit more, this is a really nice overview figure that was from a paper that Dave Siegel led. And it kind of breaks off the biological pump into 3 different types. The first is at the farthest left of this figure, the mixing pump. And this is kind of the physical transport of biologically fixed carbon from the surface down to depth. And this can happen at a variety of scales. It can happen at the largest scale, meridional overturning circulations, also things like mode water formation in certain regions of the ocean, and wind-driven Ekman subduction, kind of these large-scale events. It can happen, in things like the mixed layer pump, so these seasonal changes in the mixed layer depth, where suddenly you have a lot of carbon that used to be in the mixed layer, and in the springtime, you get abrupt restratification of the upper ocean, and it leads to a lot of carbon being brought down out of the mixed layer of the ocean. And then you have, on much smaller scales, the eddy subduction pump, and this is meso and submesoscale dynamics, acting, scales of something like 1 kilometer to maybe 100 kilometers that can inject water masses from the surface into the interior of the ocean. And again, a lot of questions here about How deep does it inject, and what does that imply for the ultimate lifetime of this carbon? You also have the gravitational pump. A lot of this is really talking about sinking particles, be they phytoplankton, be they aggregates, fecal pellets, or carcasses. And this tends to happen on something like daily to seasonal timescales. And then you have the migrant pump. And I don't think a lot of people in this session are necessarily talking about that, but it's still a very important part of the ocean carbon cycle when we talk about the biological carbon cycle, especially. And this happens a lot in kind of diel vertical migrations, but it can also happen seasonally with kind of seasonal vertical redistributions of different types of fish. So a lot of what we've been talking about, us presenters, as we're making up this this plenary session, is Stommel diagrams, and this is kind of a classic one. We're on the y-axis, you have time scales, and then the x-axis, you have horizontal spatial scales. And you know, when we think about oceanography, we have to think of time scales from seconds, or maybe even less, to hundreds of years. And we need to think of spatial scales on, millimeter scales, or maybe even smaller, all the way up to the full ocean basin. And a lot of what we end up talking about kind of ends up in this general range, phytoplankton blooms that happen over days to months, and, hundreds of meters to 100 kilometers, fronts and eddies, a lot of the other phenomena that are in here, plankton migrations. But a lot of what we're really trying to answer is up here. How do things change in the climate? How do, shifts in, decadal oscillations, or even just, seasonal cycles, how do those change the overall carbon cycle? And one of the things that we also need to think about is what observations we're using here. So I'm going to share some slides that Andrea Fassbender made for a previous OCB-funded workshop last year. And this is thinking about where a lot of our observations are. So this is the same type of Stommel diagram. You have, kind of spatial scales in the x-axis and temporal scales in the y-axis. And you see that we have a lot of, mooring observations, which make measurements at pretty small spatial scales from, minutes to decades. We have research cruises, which are often somewhat regional. We have satellites, which give us information, not at the very smallest scales, but over the entire basin. And then we have kind of our really big decadal hydrography programs, which, span decades. One of the things is that if we kind of flip this on its head and we think about the depth distribution of all of the measurements that we have, a lot of these measurements are focused at the surface. And there's, some relatively big gaps, especially when we think about subsurface measurements that we might want to be understanding. And some of these gaps are being filled in. And, we had a great presentation from Ken Johnson yesterday about the GoBGC program. And, that's part of what's filling in a lot of these gaps with the Argo program, and especially the core deep and BGC Argo floats and the deep floats that are really filling in this picture for us. But a lot of the talks that we're, going to have today, especially at the very end, are going to be discussing many new measurement strategies. So a lot of what I've been talking about so far has just been the carbon cycle in general. But one of the main reasons why we're so interested in this is because of the potential of the ocean in marine carbon dioxide removal. And there's a lot of different ways in which we can think about doing this. 5 main ones: ocean alkalinity enhancement, which a few of our speakers are going to be talking about; a macroalgal cultivation, which we're going to have one speaker session about; and ocean fertilization, we'll have some lightning talks on. The other 3 I don't think are going to come up directly here, but you might imagine that all of these different ways of thinking about MCDR are going to run up into a lot of challenges when we think about Stommel diagrams of time and space. One of the ways we can think about, one of the kind of early, this was an iron fertilization experiment, the Southern Ocean Iron Release Experiment. And, they kind of made a patch of iron addition in one small part of the Southern Ocean, south of Australia, and then about a month or so later, they could see from satellite that it had spread out into this really long, elongated filament. And you could imagine all of the challenges that might be around thinking of the spatial scales of how do you monitor that? How do you report that? How do you verify that? How do you kind of understand the broad scope of what we're doing here and measure it and quantify it? So that's kind of a brief introduction to what the session is going to be about. I'm going to turn it over to Hongjie in just a second, but I just wanted to give a quick overview of the talks and where they look on a Stommel diagram. So we're going to start off with Hongjie Wang at URI. He's going to be talking about natural carbonate variability in MCDR. We're going to move to Elizabeth Yankovsky at Yale, who's going to be giving a geophysical fluid dynamics perspective. And then Kristen is going to kind of link a lot of spatial scales together in the context of some seaweed-based climate solutions. We're also going to have a number of lightning talks in here. And then in the afternoon, Manu Prakash at Stanford is going to start us off thinking about very small scales of marine snow. Melissa Omand is going to be getting us a little bit larger with some low-cost technology to measure sinking carbon fluxes. And then our last speaker session, before we go into a bigger group discussion, is going to be Naomi Levine, who's going to be, again, kind of giving us everything from the very smallest to almost the largest scales, thinking about microbes and BGC, biogeochemical modeling. And with that, I'm going to stop sharing my slides and turn things over to Dreux to introduce our first speaker.
All right, so now we're going to have our first speaker, who is Hongjie Wang from URI, and she is hopefully going to be starting to share her slides in a moment, but she is going to be talking about natural carbonate variability and using that for monitoring MCDR.
So thank you for this intro, and good morning, everyone, by the way. I just like Zach mentioned, I will focus on the implication of the natural carbonate variability for monitoring of MCDR. I will focus on one CDR approach, ocean alkaline enhancement, because of time limitation. And so I think Anderson's question is really important, because when we compare a signal, we always want to compare a signal of MCDR to a natural variability. So that's basically the major motivation for this talk. Hopefully my talk can set up the stage for the following speaker too in this session. So I have two questions basically to guide through all this, my talk for the next 20 minutes. So the first question is, what level of natural carbonate variability do we face when targeting the ocean alkalinity enhancement? And then if we can detect the variability or the CDR signal, can we rely on ocean alkalinity enhancement to deliver a stable or CO2 removal when scaled up, based on a very simple mass balance perspective? And this is a very intro level of the inorganic carbon system. So this icon here, very cute icons, are the CO2 CO2 in the atmosphere, and about, okay, where is my? About 1/4 to 1/3 of this anthropogenic CO2 has been dissolved in the ocean. So basically move the reaction from the left to the right. By doing this, increase the hydrogen ion concentration and the bicarbonate and reduce the carbonate. And the total alkalinity, interesting, is it kind of stays stable, because the increase of the bicarbonate and decrease of the carbonate can fully cancel each other if there's no other process in this place. And so meanwhile, the increasing air CO2 actually speed up the land weathering. So I'm using the calcium silicate mineral to represent this process. Basically, the weathering of this rock can take up CO2, convert the CO2 in the form of the bicarbonate. So when this bicarbonate was transported into the ocean, it can increase the alkalinity in the ocean. And in fact, So back in 2014, STATS team actually already report the increasing alkalinity in the major U.S. rivers. The green here shows the increasing alkalinity. You can really see Mississippi River alkalinity increase in the northeast, also river alkalinity increase for majority of rivers. So this increasing of river alkalinity is a very good indicator of intensified land weathering. And so hopefully can suggesting that the future enhanced land weathering approach could be an effective strategy for the future carbon sequestration. And so I'm basically, okay, if you're new to Rhode Island, so when I first come to Rhode Island 3 years ago, I was amazed by the beautiful blue hydrangeas. And by the way, hydrangeas only be blue when the soil is acidic. So which basically means the soil of Rhode Island, New England region is very acidic. I'm sorry, my husband is yelling in the other room for another meeting. I need to turn down the dog. Okay, sorry, I'm back. So, and after joining URI about 3 years ago, I got the privilege to work with a group of excellent scientists and students. And so, and we focus on kind of using one golf cart, and which near to coastal pond, and to examine the potential enhanced land weathering. And because again, the hydrangeas show the soil is very acidic, which not only make the hydrangeas blue, but can also make the land weathering in this New England region has a relatively higher weathering rate compared to more basic soil in other regions. And so we are very lucky to work with a team, a golf cart in this right corner here. So which is 50 acres of golf course, and which sit near to this pond. And so it's very interesting for this golf cart people. They want to maintain their soil health. They routinely add the limestone to increase the pH soil because when pH is too low, this is not good for the grass. They have difficulty in absorbing nutrients. So for the routine maintenance, they add limestone to adjust the pH. And this gives us a very good study area because some like the actual limestone may deliver to the pond. Or if the enhanced land weathering take place, they can also increase alkalinity to make it more perfect. And they, for this pond, only have one outlet to this Rhode Island Sound. So basically, the simple hydrology here can make it's really make this areas a very good study to trace this alkalinity increase really to the coastal liming here. And again, by the way, there's no river to the pond. The only freshwater input is groundwater. So if there's any enhanced land weathering signal happen here because of the coastal liming, and this is a very good study to observe this. So then we got a chance to work with very wide expertise from groundwater and to like water column carbon in the system. So it's very cool to study this area. Okay, my curse. Okay, so back in last May, about 15 tons of a crushed limestone was added to this golf cart, and the limestone is not like a big stone, of course, the crust, and in the size of 1 to 2 millimeter grain, and made with 90% of calcium carbonate by mass, and the other by 10, a few percent is by other mineral. And so after the application, the soil pH increased about by 0.1, which make the golf cart very happy because by increasing the pH by 0.1, a little bit more than 0.1, and can make the soil more healthy for the grass. And so for us, for the scientist party, we are very interested about to see how the alkalinity from the groundwater responds to the surface liming. And so we sampled the groundwater and in these 3 sites, all the studies were carried by Pat and Becky and my student. And so then they measured the alkalinity. So I'm here present one alkalinity in the groundwater in this pink side. And you can really see very interestingly, alkalinity indeed increase, okay? But the increase start before the liming because this is the liming time, and the alkalinity in the groundwater start increase before the liming. And so the average alkalinity increase through this 1, 2, 3 sites is about 500 micromole per kilogram. But I have to be very clear, it's unclear to us how much of this 500 micromole per kilogram increase can be directly attributed to the coastal liming, but the groundwater alkalinity increase. Okay. And so in an ideal world, if the pond alkalinity should increase response to the groundwater alkalinity change, regardless of the reason for this alkalinity increase, okay? So assume this line represent the before liming alkalinity versus salinity regression curve, and then after liming, if the fresh water in the member increase, then the alkalinity versus salinity, the relationship should shift upwards, right? So again, no matter how much of this alkalinity increase contributed result from this coastal liming, but in real world, thing can be very interesting. So again, this shows our observation alkalinity versus salinity before liming, and then after liming. Instead of to see the blue one is before and the red ones after liming, instead of to seeing a upward shift of alkalinity versus salinity, we actually observed a downward shift of alkalinity versus salinity, which is like can only show there must be another process, some other process that can change alkalinity. So what's that? And then okay. So my student Fiona TV Kamahawi did this analysis. Okay, I won't go through the part slowly. So here shows delta alkalinity, delta DIC, which is defined by the means the alkalinity or DIC variability after take care of the water mass mixing, and then this 2 versus 1 line represents the calcium carbonate dissolution when alkalinity is positive, or calcium carbonate precipitation when the delta alkalinity is negative. And this line shows the photosynthesis when DIC is negative, or respiration when delta DIC is positive. As you can see very clearly, the alkalinity of delta alkalinity for delta DIC regression shows a strong signal of calcium carbonate dissolution when temperature is below 15 degrees. And then it switched to the precipitation when temperature warm up over 15 degree. Okay. And so we are very clear that the water column omega with the calcium carbon saturation state is still over than 1 when in the cold season. And again, my student and we did some incubation of oyster shells, and we found even for the air treatment, basically mean the omega for the water column is still over than 1. We still observed alkalinity increase. So which means the shell still dissolve in oversaturated water column, but there's no dissolution if we kill all the biologies by adding mercury chloride. So which means the microenvironment can speed up dissolution even though the bulk water column omega is higher than 1, which can explain why there's a strong calcium carbonate dissolution in the cold season. And then in summer, it turned out the pond is a very successful oyster aquaculture. They harvest oyster shells, oysters, and sell them in summer and tourist season. It's very cool. And so then the strong biogenic calcification here, I'm only use the oyster to represent them. Of course, there's some clams, the other like bivalves there. So they remove a significant of alkalinity. So in warm time, and coincidentally, this warm time is after the liming, after May, when the water temperature got warm up. So what I want to say is the presence of the biogenic calcification in the summer or dissolution maybe over the annually, and can alter the carbonate system and can mask ocean alkalinity enhancement signal. So then one of the lesson we learned from this Winnipeg Pond experiment is with more than 1 years of high resolution alkaline sampling weekly and monthly, revealed that natural variability, alkaline variability contribute result from the calcium carbonate cycle is about 77 micromole per kilogram, 77. And so then this question triggered me to ask, can we expect a similar variability as well, or are there quiet zones more favorable for ocean alkaline enhancement detection? To answer this question, I worked with my former graduate student, Thomas Chiara, who now is instructor at the Coastal Guard Academy. And so we went to the dataset, CODAP North America dataset, and so we did a very similar analysis to again tease out the alkaline calcium carbonate by using these equations. If you are interested by equation, we can talk about details later because the time limitation. But the core point, the principle is here, is like for the observed alkalinity, we adjust the water mass mixing and also adjust the biological net ecosystem metabolism by using AOU as a proxy and N star and using to represent the nitrogen fixation or denatrification. And then we define the variability of the alkaline calcium carbonate again due the symbol of alkalinity calcium carbonate is due to the alkaline chains due to the calcium carbonate process. Okay, we define as 80% of data range. It's not like the maximum minus the minimum, but it's the middle 80% of data range to avoid the outliers. And then we constrain the analysis in the top 100 meter. And so whenever we talk about variability, the data coverage is a big issue, right? So to avoid kind of the uneven data coverage, we only focus on the grid, 5x5 grid actually, with more than 7 unique months in each grid. It turned out with this kind of requirement, only 1, 2, 3, 4, 5 grid and can meet our requirement. And then we add the hot and the bed, sorry, move a little bit, and these two sides for comparison. And so then in Rhode Island here, Winnipeg Pond, so again, here's alkalinity calcium carbonate against the temperature. When temperature warm, it's very warm, precipitation, shelf formation, remove the alkalinity, cold-station, dissolution dominate, increase alkalinity calcium carbonate. So this is Winnipeg Pond. And we saw a similar pattern in outside of this Florida, but we didn't see this trend, this relationship between alkalinity, calcium carbonate against temperature in the Bering Sea. And so the one key message here is the alkalinity variability due to the calcium carbonate cycle range from about 20 micromole to 77 here. So that's the range. And meanwhile, how much should alkaline increase to remove 1 petagram carbon per year if we rely on ocean alkaline enhancement? So I did a very simple calculation. So assume mixed depth is 100 meters, which is definitely not right, but reasonable good. And then assume the global ocean average is 360 million square meter, and assume 100% OAE efficiency, which shouldn't be that's ideal. And then the needed alkalinity is about 100 paramole per year to remove 1 petagram carbon per year, so OAE. And then that means the alkaline concentration increases only 2.8 micromole per kilogram per year. So this is the expected alkalinity increase with a meaningful OAE for climate mitigation. And then so if you compare the 2.8 against the net variability range from 20 to 77, this is very small. And to make even more challenging, the detection of OAE signal more challenging, so again, I plot the alkalinity calcium carbonate against the time in that 5 grids, which has reasonable good data coverage. You don't really see long-term change, but if I put this Hall station, and you can start to see a second increase of alkalinity, but with just past the significant test. Okay, so here is a bat. You can really see the bat alkalinity has significant increase with 0.34 micromole per kilogram per year. And the change of alkalinity over time, it's basically it's very consistent with the recent publication by Barrett et al. 2025 based on GLODAP version 2, and they found alkalinity in some ocean area, especially in North Atlantic, increase. The increased read is about 0.1 micromole per kilogram per year based on another kind of independent method. So what I want to say is like, so the long-term trend in alkalinity calcium carbonate already start to show in some open ocean region. So this can only make the detection of potential OAE signal even more challenging, because how will we separate the natural increasing trend from the OAE signal, right? Which is supposed to be very small anyway. Okay. Okay. So I think I still have time to go to my second question. So, okay, if we have all the technology that can detect that small variability, but hold on, whether the OAE can deliver a stable CO2 removal when scaled up. So this is some ongoing work in my lab, but I think that I would still want to share with you. If this is, again, go back to the intro slides, like, and so when we say we want to add alkalinity, let's assume this, I use sodium hydroxide as represent alkaline material because it's simple. And so when we want, hopefully we add the alkaline to remove the hydrogen ion and shift the reaction from the left to right. This process can significantly increase the carbonate. When the carbonate concentration is too high, and so it will trigger secondary precipitation, which is abiotic precipitation. So here's a lab demo I did. So basically, this is the natural seawater, and I add sodium hydroxide. You can really see the cloudy formation. This is the abiotic precipitation, and the water become cloudy over time. And so that's again calcium carbonate precipitation, and this is easy to detect. And of course, there's some publication related to this to study the stability of this added alkalinity if you add too much alkalinity. And we kind of straightforward. You don't want to add too much alkalinity and to avoid the leakage of the alkaline materials. But what I want to brought your attention is something more subtle. Let's say I use this chlorophyllus as a representative plankton calcifier. So they may also respond to the added alkalinity in the open ocean. The reason I know there's a lot of going on work in Mexico, some maybe lab work to study the response of these calcifiers to the increasing alkalinity. And so I think I want to put this the importance of this potential response of plankton calcifiers in the global alkalinity budget. So here I kind of modified the alkalinity budget based on the recent publications on the and the major alkalinity source or remove a sink. Basically, the in the open ocean, there's a net PIC, particularly in organic carbon with calcium carbonate production. And the production, this is in teramole per year. So the production of the PIC is about on the magnitude of 1 petagram carbon per year. So net PIC production, then some net calcium carbonate burial. And so then but overall, because of the alkalinity production, so the groundwater discharge or anaerobic respiration and river input, more or less, the ocean alkalinity is more or less mass balance. Okay, the source is 66 and the sink is 60 teramole per year. So that means overall, the ocean jungling between source and sink more or less balance. And the production of this plankton and gasifiers remove alkalinity on the magnitude about 133 based on the publication thermal per carbon per year. So if we want to add alkalinity about 100 thermal per year, as what we discussed previously, the amount of this alkalinity addition is almost the same magnitude of the natural PIC export. So how will all this natural alkalinity removal response to such a huge amount of alkalinity addition? I think it really deserves a more in-depth study from an ecosystem perspective, maybe rely on the modeling and even the lab incubation, macrocosm incubation. So hopefully we can really understand how the ocean alkaline responds to the huge amount of alkalinity addition in the future if we want to rely on OEE as a reliable carbon sequestration approach. Because again, the perturbator signal is too large. It's relatively big compared to natural process. Okay. How do you stop wiggling? Okay, so with this one, I want to summarize my key points of my talk. The first, the natural alkalinity variability due to the calcium carbon cycle range from about 20 in the open ocean to about 80 micromole per kilogram in the coastal area. So this signal is definitely higher than expected. The meaningful OEE-driven signal was 2 to 3. So when I say the meaningful OEE is to remove the carbon paragram carbon per year. And then observed alkalinity increase in the open ocean could result from the reduced precipitation or enhanced dissolution. And distinguishing this future OAE signal from this natural increasing will be challenging, I want to say. And then how might the OEE alter the nature calcium carbon feedback loop over what time scale when scaled up? I think it deserves more studies. And so with this, I want to acknowledge my team, the Winnie-Parker Pond team, and my lab, and Noah's lab from Yale to help to analyze the soil result. Thank you. With this, I can take questions. I don't know how much time I use.
Hi. So unfortunately, we're not going to have time for questions. We have to move to the next speaker. But any questions that you guys have for Hongjie, please put them in the chat. If they're broader questions, we might come back to them in the discussion. But thank you so much. That was a wonderful talk. And now, Elizabeth, can you start sharing your slides? I recommend if you've got a cell phone handy, put your timer on. It'll help you keep track of the 20 minutes for you, and then which will allow some time for questions. But now we have Elizabeth's slides up. So Elizabeth Yankovsky is speaking to us from Yale University, and she's going to talk about geophysical fluid dynamics and MCDR, which is pretty exciting stuff. So take it away, Elizabeth.
Thank you very much for the introduction. And I also want to acknowledge that a lot of the work that I'll be presenting today was also done with some of my collaborators at Seaworthy. So yeah, I'll be speaking about geophysical fluid dynamics perspectives and the interplay with the carbon cycle, as well as perspectives on marine carbon dioxide removal. And a really large theme underlying a lot of the talks today is really the range of scales and the nonlinear interactions between all of these scales, which poses a really large challenge for us in understanding this really complex Earth system. So I'll be speaking a little bit more with a focus on kind of the dynamics governing the atmosphere and the ocean and how that impacts the carbon cycle and how that kind of sets really significant implications for our ability to do carbon dioxide removal and really quantify those efforts. So when we're thinking about carbon dioxide removal, we really have to kind of address the range of scales and really think about how to responsibly do carbon crediting for these efforts, which means that we have to understand how these interventions are going to evolve on scales that range from millimeters to thousands of kilometers and timescales of potentially millennia. So to give a brief overview, I know this has been covered slightly in the past two talks. a brief overview of ocean alkalinity enhancement. So the idea is that we're adding some sort of alkalinity to the surface ocean. Here it's depicted as a hydroxide ion. And the idea is that this induces a deficit in the partial pressure of CO2 in the surface ocean, which then causes a flux of atmospheric CO2 into the surface ocean. The hope is that over time, this atmospheric CO2 will be permanently stored in the ocean as bicarbonate and carbonate ions. And so the way that this exchange of atmospheric CO2 going into the surface ocean happens is through wind-driven gas transfer. And so there are two really important aspects to take away from this slide. The first one is that in order for these carbon dioxide removal efforts to be effective, the alkalinity must remain at the surface. Otherwise, there will be no kind of deficit of partial pressure of CO2 that will cause this flux into the ocean. So of course, for alkalinity to remain at the surface, this can be really modulated by a lot of the mixing and a lot of the physical dynamics that are happening in the highly dynamic ocean mixed layer. So there are a lot of instabilities, waves, and turbulence that evolve in this part of the ocean, and they can really fundamentally influence some of the vertical transport processes that occur and potentially move alkalinity out of that surface mixed layer and deeper into the ocean, which would cause the effect of the carbon dioxide removal to be minimized. The other really important piece, again, is that wind is what's driving this gas transfer. So the atmospheric CO2 kind of getting incorporated into the ocean. The way that this is often represented in a kind of simplified manner is that the flux of CO2 into the ocean is a function of these three variables. So delta PCO2 is the change in the partial pressure of CO2 between the atmosphere and the ocean. So again, this is kind of reflecting the deficit of PCO2 that we see that causes this flux into the ocean. We also have the solubility of CO2 in seawater. And finally, we have this K, which is the gas transfer velocity, which is kind of a vertical piston velocity that is indicating how rapidly the CO2 is getting incorporated into the surface ocean. And the really key piece to take away from this is that this is often parameterized or represented as this kind of stress form, where there's a coefficient, and then U squared, where U here is the 10-meter wind magnitude. So in other words, this is a quadratic relationship with the wind. And so the key piece to take away from this is that we really have to understand both the wind dynamics and the ocean dynamics, as well as the interplay of how wind may influence convergence and divergence and other instabilities and processes in the surface ocean. So this brings us to why kind of understanding all of those dynamics is quite challenging. So GFD from the ocean perspective is challenging, of course, because it evolves on so many different scales, and all of these scales are non-linearly interacting with each other. We have processes on the order of millimeters and subseconds, kind of governing air-sea gas exchange through bubbles and wave dynamics. We have kind of larger scale eddies and waves that may influence stirring of tracers and also really fundamentally shape the vertical transport, as I mentioned, out of the mixed layer and out of the surface ocean. We have things like tides. Then on larger scales, we have a lot of climate variability, such as El Nino, the wind-driven ocean circulation, and then even on the kind of scale of tens of thousands of kilometers and thousands of years, we have orbital variations that really may also fundamentally shape how the ocean and atmosphere are evolving and, of course, impact the carbon cycle there. So one thing to note here is that this is just the ocean. And so when we're thinking about carbon dioxide removal, we're also thinking about the interplay of the ocean dynamics with the carbon cycle and the atmospheric dynamics, and also the biogeochemistry and the lithosphere even. So there are a lot of Earth system components that are really coming together to shape how the carbon cycle looks like and how it evolves on these scales of tens of thousands of years. And so when we're thinking about carbon crediting and really doing carbon dioxide removal in a responsible way, we really have to address the quantification of how that CO2 will evolve over all of these scales. And of course, that's an extremely challenging problem and one that we can't really use simply one tool to solve. So leading into kind of the next part of this is that models represent our best understanding of all of these complex Earth system interactions. Models have come a really long way since they were first developed in the 60s and 70s. And nowadays, the idea is that we're solving kind of all of these equations governing these various Earth system components on some sort of discretized grid. And it's really encapsulating kind of all of the interactions between these various constituents of the Earth system. Models do have limitations, but they've also had great success, as evidenced by the Nobel Prize in Physics going to some of the earliest climate modeling efforts. So this is an example showing some of what models can capture. This is the community Earth system model developed at NCAR. So the idea here is that there are a lot of different constituents that go into this model. The kind of core ones are the ocean component, the atmosphere component, the land component, and then of course there's chemistry and sea ice and land ice as well. There's a lot of really amazing development that's being done both at NCAR and other labs around the world that really tries to push forward these models' ability to try to simulate all of these complex aspects of the Earth system. So just to give you a couple of snapshots, we have really interesting kind of developments in land models. Again, this is from the CESM developed at NCAR. So some of these land models are quite complex in that they permit a lot of different types of surface representation for different types of trees or different types of land kind of surface cover. We have very complex biogeochemistry models being developed. Some of them even incorporate things like fish dynamics, which is pretty exciting. And then, of course, a lot of aspects trying to understand how different fluxes and different parts of these systems are all interacting with each other. So in general, you can envision that these models can potentially do a really great job representing some of the complexities of the carbon cycle and the interplay of various geophysical processes across all of these different scales. But of course, there are still a lot of limitations. And so in the next part of this talk, I'll speak a little bit about how we can leverage models to address CDR efforts, particularly focusing on trying to understand the evolution across all of these different scales. And then I'll speak a little bit about what are the challenges as well. So a big part of carbon dioxide removal is trying to kind of make sure that it is safe and verifiable. And this involves measurement, reporting, and verification. That's being done in a consistent way across a lot of these marine carbon dioxide removal efforts. But of course, there are really significant challenges, as also alluded to in the previous talk, in terms of being able to measure some of these interventions. One of the big challenges is that these interventions, even when they're kind of initiated at small scales, very rapidly propagate to pretty large scales. So this is showing an ocean alkalinity injection off the coast of Africa after a year and after 15 years. So you can see that this alkalinity and this corresponding surface PCO2 deficit has basically spread over the entire Atlantic basin. And so trying to observationally measure this is quite challenging. And even in models, you really need to run kind of multiple scenarios with counterfactual baselines to really assess the role of a particular intervention. And of course, trying to accurately resolve all of these different scales, even in a model, is a big challenge. Another challenge kind of that really motivates the need for models, this is a movie by Matt Long, that's seaworthy, is the fact that there are very large fluxes of CO2 in the background. So this is showing the air-sea CO2 flux, where positive means out of the ocean, so outgassing of CO2, and negative means into the ocean. You can see that there's a really complex pattern all around the globe here. And so when we do ocean alkalinity enhancement or other CDR efforts, the magnitude of those CO2 fluxes that those efforts initiate is usually very small relative to this background variability. And so that makes it even more challenging in terms of being able to kind of model, observe, and really verify over long time scales what effects these CDR interventions will have. So kind of bringing it back a little bit to marine carbon dioxide removal. Again, this is a multi-scale problem that's very fundamentally shaped by the background geophysical fluid dynamics that we see in the ocean and in the atmosphere. So when we think about ocean alkalinity enhancement or other marine CDR efforts, they typically happen on spatial scales of order 10 to maybe hundreds of meters. And of course, those efforts are going to be really shaped by underlying fluid dynamic properties that are occurring on millimeter scales, all the way up to kind of global basin scales. And so how to try to capture all of this in a single model or in a single framework is a big challenge. And in my next two slides, I'll show two examples of both microscale and global scale dynamics. So in terms of an example of how geophysical fluid dynamics on microscales can be really fundamental to understanding carbon dioxide removal. This is a really interesting study published by Yang and Timmermans in 2024. And the idea is that they were assessing the settling of mineral particles, which is one way to do ocean alkalinity enhancement, with application to ocean-based CDR. So here the idea is that typically the way that we predict how particles settle in the ocean is through something called Stokes settling, which basically gives you some sort of characteristic velocity at which these particles will sink. This is really important because you want these particles to stay in the surface ocean long enough to really dissolve and also to create the CO2 uptake. But the problem is that when you actually have a sheared and stratified fluid, it can develop really interesting instabilities. And in this study, they were able to actually resolve these instabilities. And they found that when you actually resolve these instabilities, the flux of kind of the downward velocity is roughly an order of magnitude more significant than just the Stokes settling velocity alone. And so this is really problematic because that means the efficiency of these OAE efforts is going to be much smaller than we envision if we actually take into account the fluid dynamics that's happening there. To go to the other scale, this is an example of a study that looked at the global dynamics. So this is using a roughly 1 degree ocean model. So the grid size is 100 by 100 kilometers here. And the idea is to really look at doing ocean alkalinity enhancement in all of these different polygons all around the globe. And so due to the fact that this is a pretty coarse model, we're able to actually predict how ocean alkalinity enhancement will evolve and look at the actual efficiency that we see. So higher number means higher efficiency of the ocean alkalinity enhancement over the entire globe. And so this is really powerful, but of course here the challenge is that we're really missing a lot of crucial dynamics in this model. And so during my time at Seaworthy, we really kind of thought about regional models as being a solution that really bridges some of the small scale dynamics with some of the global dynamics and really tries to focus in on really capturing these kind of intermediate scales very well. So this is some work that that I've been kind of still in progress for me, but this is looking at regional models to try to understand the role of resolution and various scales of ocean turbulence on carbon uptake. And also to try to unify kind of various modeling efforts, thinking about different groups that might be running models at different resolutions. So to try to see if there's some sort of systematic error that we might be seeing when we run a coarse model compared to a fine resolution model. And just to give you some kind of preliminary results from this, this is showing videos of these 3 different resolutions. Here, there's an alkalinity plume that's spreading. You can see in the higher resolution case that we do have more fronts and filaments and fine scale dynamics being resolved compared to this coarser case. But in terms of looking at some of the properties of where the alkalinity is, we see that the alkalinity that's remaining at the surface is somewhat smaller in the high resolution case. This is perhaps because we're starting to resolve some of the submesoscale dynamics, which are known to potentially initiate more vertical motion. But in terms of the actual uptake efficiency that we see in the end, the high resolution case is only marginally smaller. And this is perhaps because the resolution difference between these isn't that significant, and we're not really resolving submesoscales here. So I'm kind of doing some ongoing simulations to look at 1 and 2 kilometer resolutions to really see if we see kind of a bigger downward trend in the uptake efficiency when we resolve submesoscales to a greater degree. Just to give you a little bit more comparison between the coarsest and the highest resolution here. In this figure on the bottom, I'm showing the actual ratio of the alkalinity that at the surface between the high resolution and the low resolution case. So anything that's less than 1 here means that the high resolution case has less alkalinity at the surface. And indeed, throughout most of the simulation, we see that the high resolution case has about 80 to 90% of the alkalinity in the surface that the lower resolution has. So it would be interesting to continue this and see if, as we move to the higher resolutions, this will continue to go down. Another interesting thing, if we look at the actual plume and kind of looking at the center of mass of where the alkalinity is in the vertical, we can see that in the higher resolution case, these fronts and filaments that we're resolving, they tend to be actually where we have the deeper alkalinity and where we have the subduction. So there's definitely some preliminary evidence that is suggesting that these submesoscales kind of frontal dynamics are really driving more vertical motion and more vertical transport. But of course, there are still a lot of challenges with these regional models. A big one is that alkalinity leaves the domain pretty rapidly. So as I showed in the kind of larger scale modeling slide, the alkalinity typically leaves the domain within a couple of years and then propagates to basin and global scales over decadal timescales. And so this is still a challenge for really looking at the durability of these CO2 removal efforts because we really are not able to see the longer term time evolution of this. Another big problem is that in all of these simulations, we're relying on low resolution atmospheric forcing. So this is typically using atmospheric reanalysis products like JRA, where the atmospheric forcing is on a 25 kilometer or so grid. And so when we think about the air-sea gas exchange being driven by the velocity of the wind squared, that could be a really big problem because if you have really small gusts or really small kind of wind events that are potentially much stronger than these averaged out smoothed wind fields, we could be really missing a lot of the important air-sea gas exchange and also a lot of the kind of wind-driven ocean dynamics that may evolve. The other piece is that resolving submesoscale turbulence is still very expensive, even for these regional models. These models for a 1 or 2 kilometer simulation will take weeks to run. And finally, there's still a lot of variability. So this is showing kind of a comparison of different years. So these are all January releases of alkalinity, but you can see depending on the year, the actual flow field looks quite different, and the resulting megatons of CO2 removed also really substantially varies year to year. So this kind of methodology of using hindcast simulations where we just do one experiment and then use that as our basis for CDR quantification, it's really flawed. We still need really bespoke simulations for a given scenario to assess the impacts of the CDR. And so something that I've been thinking about more recently and would love to hear thoughts and potentially any people interested in collaborating on this is how can we leverage recent modeling and also AI developments to tackle the ocean-based CDR quantification challenge. So this is showing a recently published paper from 2025 where they're presenting this model called Oceananigans. So this is a GPU optimized model, and it's a really kind of bare bones model, in my opinion, compared to some of these traditional Earth system models. It really does away with a lot of the parameterizations that Earth system models typically have and really relies purely on the numerics and really tries to target just really intense computing speeds. So in doing so, they were able to achieve pretty remarkable speed ups. So kind of running on 64 GPUs. This core can simulate a decade of ocean dynamics in 1 day for an 8 kilometer horizontal resolution, which is pretty remarkable. And also, there are some really interesting developments on the atmospheric side. So we know that AI has had a lot of success in weather prediction and weather forecasting. And so this is an example of a tool developed by NVIDIA. It's called the Generative Correction Diffusion Model. And what this tool does is that it basically downscales atmospheric products in order to generate very high resolution winds. And so one thought that I've been kind of thinking about pursuing is to try to couple something like the Oceananigans GPU optimized model to something where we derive these very high resolution wind products from these kind of AI tools. And so this is something that I'm kind of currently exploring, trying to implement together in order to really improve our ability to predict these CDR interventions. So rather than just relying on kind of regional or coarse global models to try to kind of leverage these weather prediction AI successes in order to force these very high resolution GPU optimized models. Okay, so I think I'm running out of time, so I'll leave my summary up, and we'll be happy to take questions.
Thanks, Elizabeth. That was great. We do have some time for questions, so if people want to put those questions in the chat, I forgot to check, May, whether or not people can actually unmute to ask questions. We'd love it if there are any early career questions out there, but we have a question in the chat from Shoily. How do you deal with extreme storm events?
Yeah, that's a great question. So as of now, a lot of the CDR quantification efforts are really treated as hindcasts, where we kind of run things like this ocean atlas for alkalinity enhancement, where we just take past scenarios and kind of climatological forcing, and then study in different regions of the ocean, how does uptake happen. Some of these regional ocean modeling efforts take it a little bit further in terms of actually looking at a very high resolution specific area, but nonetheless, there isn't really this forecasting aspect that has yet been implemented, and storm events aren't really dealt with. So I think a lot of the assessment just kind of comes from hindcasts and past simulations. So I think dealing with storm events is really important, and understanding how high frequency atmospheric variability may impact the uptake of carbon really needs to be done.
All right, we do have time for at least another question.
Yeah, thank you for the great talk. And I have one questions about this additionalities on that, because you calculated additionalities like between these two experiments, like the OAE and control simulations. And I have a really good questions about this, how to make them baseline for the control simulations. So for example, we can do like the climatology of the 30 years, and then like considering all the seasonal or dino cycles. So do you have any idea? But I think that this additionality would be really depending on this baseline. So how do you think that this calculating baselines on here?
Yeah, I think for calculating baselines, you really have to have an identical flow that's in your perturbed and not perturbed case. So in my simulations, I've been using Marble, which is a biogeochemistry library, which solves two carbonate chemistry systems at once. So all of the ocean dynamics are the same between the control and the perturbed case, and it's only the alkalinity perturbation that's changing. So does that answer your question?
So you mean that? Yeah, so you mean that you calculated the differences for the each day's differences, you mean? Or just calculating, not the climatologies,
Yeah, at each time step, you can calculate the differences between the two.
I see. Yeah, thank you.
Thank you.
So we're gonna move to the lightning talks.
All right, good morning. So my name is Monique Messier, I'm at MBARI, and I will be briefly talking about how some variability in the deep ocean may be driven by what happens at the surface, and in particular, ocean circulation and upwelling. So this study was kind of initiated by looking at that time series that you see here on the bottom left, which is a time series of deep sea POC fluxes collected at 3,400 meter depths off of the California coast at Station M. And so as you can see, there's a lot of very high pulses that have been increasing in magnitude and frequency over time. And so we've been trying to understand what creates such high variability in the abyss. This is just a different view of the same time series. On the top, you have the deep sea POC fluxes. The time series below is a time series of export as predicted from ocean color. It's just proportional to satellite primary production. And you can see that it's a lot less variable. We actually get the background pretty well, which is a point of the previous slide I forgot to mention, but we kind of miss those high pulses. We see a very much reduced variability relative to what happens at the bottom. On the right, you have kind of a zoom on 1 year with a month that happened just before one of those pulses. And we have a pretty low percentage of variance explained, around 18%. Part of it is that there's a lot of other factors like what happens in the ocean interior. So that explains part of that. And so the point of this talk is to talk about a different type of product that I'll talk about in a minute. That is a Lagrangian model that's intended to represent zooplankton instead of looking at phytoplankton. And you can see that the time series that it predicts is a lot more variable, and we do get some high pulses. The percent of variance explained is still relatively low, but the interesting part about it is that if you combine the two with just a linear regression, you actually can improve the percent of variance explained by surface export by 33%. So like we think that that product has some promise in terms of helping us understand better how export it can be predicted from satellites. So just to explain a little bit how that product works. So it's a satellite-derived model that looks at the evolution of biology along current trajectories initiated at the coast. So you have a simple plankton model that is initiated by nitrate supply at time 0, which is the time series at the top. And so you have kind of that export that is generated by zooplankton in the model that peaks after about 3 weeks and is more intense when upwelling at the coast is higher. And then you take the result of that model and you plug it onto Lagrangian trajectories, and this is what the map shows. And so from all of those trajectories, you can construct daily and then monthly maps. So that model only uses upwelling, advection, and that timeline of 3 weeks. And this is just a quick validation. We use the CCELTR dataset of sediment traps. So those are at the base of the euphotic zone. And so on the right, you have a comparison of that in situ export with the export predicted from that NPZ model along trajectories. Those are backward trajectories. So basically, we go back to where the water masses originated at the coast. We run the model using that nutrient supply, and we compare it to the in situ product. So we can actually explain about 70% of the variability in surface export by that combination of upwelling, advection, and 3 weeks lag. And so just to recap the key points of what I want you to take away from, phytoplankton and export are offset in time and space, and that's because a lot of the gravitational export is also driven by zooplankton, and that's particularly problematic in places like the California current that can be really dynamic with strong advection. So that means that we can't just use ocean color to predict export from space. And using a model such as this one, it's intended to represent zooplankton, and it uses upwelling and advection, exhibits some significant skills relative to ocean color. So that's kind of a path forward to improving export from space. And basically, we think that variable upwelling and oceanic circulation can create patchiness at the surface that can drive some of those abyssal carbon pulses. And that's all I have. Thank you.
Yeah, so my name is Mara Freilich. I'm an assistant professor at Brown University, and I'll be talking about some recent work we've been doing to establish the role of submesoscale dynamics in shaping both biological communities and biogeochemical fluxes. And this is work that I've done in collaboration with a number of people, but including a PhD student, Elise Boudin at Brown. We have an understanding now that tight physical biological coupling at submesoscale has implications for both biological community structure and biogeochemistry. You heard in the introduction that submesoscale motions connect surface and interior with implications for carbon export. One thing that's critical that we outlined in our paper published in 2024 is that these motions transport whole microbial communities on timescales on which the microbial communities can respond to their changing environment. And so there's critical ecological component to these biogeochemical fluxes. One outstanding question, I think, is what is the relative role of biological and physical processes in biogeochemical fluxes? And so if we look at the cover image, this is a satellite image from the California current system, where we have this paradigm of high productivity near shore and then an oligotrophic open ocean. But there are submesoscale hotspots of biological productivity. And our question is, as these are transported by physical dynamics and have inherent submesoscale dynamics themselves, in what way are biological responses mediating the biogeochemical transport? building up a conceptual model where biology interacts closely with mixing. And so I was fortunate to collaborate with the NASA Submesoscale Ocean Dynamics Experiment, or SMODE, where we were able to use both a range of in situ, including classical shipboard observations, as well as autonomous observations, and airborne observations that gave us hyperspectral ocean color, as well as direct velocity observations from multiple platforms at submesoscale. Here's just a sample of the results. And so on the right-hand side, you can see vorticity from plane-based observations. This is not a model. And I outlined one eddy in a box. And you can see that eddy on the left-hand side in ocean color. And the blue dots are showing where we sampled the biological community composition. We have a range of genomics observations that we're using to study the ecology of these features. I won't have time to discuss those here. But you see that this feature evolves in time, but also has a tight relationship with the physical flow field and vorticity. And so here's a composite of that eddy, which we were able to observe over a week here on the left-hand side. So I'm showing vorticity again. Note the scales. This is a true submesoscale eddy, so about 10 kilometers across. And it was one of those eddies that I showed in our ocean color image. So it's an offshore, high chlorophyll eddy in an oligotrophic environment. The colored dots are showing rates of change of chlorophyll. These are Lagrangian rates of change computed using drifters. Using our direct velocity observations, we also computed vertical fluxes. Because we're able to estimate vertical velocity from the velocity observations, and we used incubation experiments to estimate growth and grazing rates. So looking at this figure, what we see is that there's a patchy increase in greens as well as decrease within the vortex core within this boundary denoted in yellow. And there's a fairly uniform decrease outside the vortex core. What we see is that these rates of change of chlorophyll, this eddy, although it's a cyclone, it was collapsing as it moved offshore. So there's a downward advection within the cyclone. But it seems to be serving as a sort of stew pot or incubator where there's active biological dynamics that are where the growth and grazing from our incubation experiment seem to account for the magnitude of the in situ estimates of loss and growth within this vortex core. And so here's an eddy. It was propagating offshore at about 6.5 kilometers per day. And so although we say that it's primarily biological dynamics causing the change within this feature, this whole feature is moving offshore, transporting coastal like conditions offshore in a submesoscale context. The vertical transport is not trivial. It's about 10 meters per day. And so we'll be publishing these observational results, including genomics, and you can see the implications for modeling in a recent paper as well.
Okay, thanks. I'm John Lauderdale. I'm a research scientist at MIT, and this paper is in close collaboration with a couple of people from University of Quebec at Rimouski. Sandy Gregorio is the first author on the paper due to some heroic modeling and diagnostics that she's been working on over the last couple of years. We're interested in what causes the change in atmospheric CO2 over the last Quaternary glaciations. So a lot of studies have kind of taken this approach where you run a model, an ocean biogeochemistry model, and you look at the different pools that the ocean carbon has resides in, where the atmospheric CO2 uptake goes. So we use an ocean model. It's kind of a simple two basin configuration, but that allows us to run it really efficiently. Don't let the simplicity fool you. It's got a full BGC model in it, and it reproduces the sort of interglacial pre-industrial distribution of nutrients and carbon. We do a carbon cycle decomposition, which is something that's a relatively new but increasingly common set of diagnostics to run. And we break it down, first aggregate it into carbon changes due to physics and carbon changes due to biogeochemical processes. And what we do is we run a full glacial model. You can see that the full glacial model gets about 40 parts per million carbon uptake. A lot of that resides in the BGC carbon pool, which includes the soft tissue pump, some changes in solubility due to alkalinity, and a biological disequilibrium term. disequilibrium is basically the deficit or the excess carbon that water mass holds when it's exported from the surface. So it's out of equilibrium with the atmosphere. What we do is then take different components of these models and run them individually. So for example, we take just the glacial temperature, the cooling, and run that in a model and look at how that changes the carbon pools. And what you can see is that the glacial temperature run achieves almost 40 parts per million. So that's the bulk of the full glacial. But if you look at this simple carbon decomposition, you can see that it's actually for completely the wrong reasons. The majority of the change is in the physical carbon pools, not the biogeochemistry. So, you know, if you were to look at just the atmospheric CO2, you might think, oh yeah, job done, it's all due to temperature. But if you look at the carbon breakdown, you can see that's for the wrong reason. We also see a very large increase in sea ice and a reconfiguration of the glacial overturning circulation. If you were to look at those individually, you would see that they create a small outgassing of CO2. So you might be tempted to write those off as not the major driver. However, if we run an experiment where we have both glacial sea ice and overturning circulation, you can see that we generate this large increase in the biogeochemical carbon storage. Next slide, please. I tried to change it myself just then. We can plot the detailed carbon pools in this kind of fingerprint style plot. And what you can see is, again, that the full glacial fingerprint of ocean carbon storage is really, really set by the glacial sea ice simultaneously run with the change in overturning circulation. If you look, you can see a large tail and sort of the southwest corner. That's the biological disequilibrium pump that I talked about. Basically the upwelling of soft tissue carbon that doesn't fully outgas in the Southern Ocean and is then resubducted. This is critically driven by the surface residence time in the Southern Ocean, which we've calculated based on some theoretical arguments from Ito and Follows. You can see on the very right of that plot, 6-year residence time in the interglacial exponentially decreases as you get through the glacial MOC and the glacial sea ice. And eventually you get this really large increase in biological disequilibrium once you get to a residence time below about 1 year. So this is really what's happening. You get the soft tissue uptake, biological pump gets the carbon into the deep ocean, that upwells in the surface Southern Ocean, but gets trapped by this combination of changing sea ice and circulation that prevents full outgassing. And those waters are then resubducted into the ocean interior physically as the disequilibrium pool. What's really exciting is that there's some C13 papers that have recently come out. Schmittner and Philman did a modeling study, OMTA did a proxy study, and they show a similar signal. Thank you very much.
Hi, my name is Heather Kim, and I'm a faculty member at Hui. I'm really excited to share this work, which I've been leading alongside an amazing team of scientists working across scales, from systems biology to global ocean modeling. So this project is part of the NSF Center for Chemical Occurrences of Microbial Planet, which brings together expertise in metabolism, biogeochemistry, and earth system science. Heterotrophic marine bacteria, or bacteria here, are crucial in the ocean carbon cycle through their interactions with organic matter and other plankton. They consume DOC, much of which is respired back to CO2, making them as a sink for DOC and a source for DOC and source for CO2. Bacteria also recycle nutrients that support other plankton, contributing to carbon export. The figures at top right show results from our recent study, one of the first to explore how bacterial respiration may change under the future climate using an Earth system model with explicit bacterial dynamics. While our study was a first century scale analysis of bacterial change, more work is needed to fully understand their impact on global carbon cycling. Modeling bacterial dynamics at global scale is, however, challenging because their microscopic processes involve high diversity and metabolic flexibility, and they interact with a complex pool of DOC that most models don't resolve well at high resolution. Therefore, these factors are typically omitted or oversimplified in Earth system models, and addressing these challenges requires a thoughtful integration of cellular scale data and models, including omics, into these global scale models. Our one-dimensional biogeochemical model shown here captures bacterial feedbacks on carbon cycling at the Bermuda Atlantic time series study or BAT site. So this BATS model resolves bacteria, phytoplankton, zooplankton, nutrients, and dissolved and particulate organic matter dynamics. This model simulates the BATS data to optimize a subset of parameters, including bacterial maximum growth and respiration rates, reducing uncertainty and improving the model scale. The model is driven by mixed layer depth, temperature, light, vertical mixing, and is built in Fortran and interfaces with three-dimensional ocean circulation models. The figures on the right show results from our baseline simulations under 30-year climatological forcing and illustrate how bacterial respiration influences ocean carbon cycling at the BAT site. The top plot show annual integrated net community production, net primary production, and heterotrophic respiration in the upper 150 meters. These results highlight that bacteria contribute over 80% of heterotrophic respiration, making them the dominant driver of these key carbon cycle processes. The middle plot shows future changes in bacterial respiration driven by ocean warming, increased stratification, and changes in DOM. The bottom plot shows, The bottom plot shows how this in turn can lead to changes in the community production and therefore carbon export potential. The results from the previous slide highlight bacterial feedbacks on carbon cycling and how those may change under future climate change. While this work is focused on the BAT site for now, our approach lays the foundation for broader investigations of bacterial impacts on the global carbon cycle. To do this, we've been developing a coupled FBA BATS model platform connected by a translator. FBA, which is flux balance analysis, is a genome scale modeling approach that predicts microbial rates by optimizing intracellular metabolic fluxes under given environmental constraints. The translator, the red box here in the middle, connecting FBA and BATS models, feeds DOM exudation rates from phytoplankton from the BATS model, and also fine-grained label DOM into specific substrates as inputs to the FBA model. This allows the FBA model to compute maximum bacterial growth and substrate uptake rates based on changing environmental conditions, including temperature, substrate composition, and availability. And these rates are passed back to the BATS model, where they now drive emergent bacterial dynamics, replacing poorly constrained parameters and equations even in the existing formulations. So we have implemented this in our minimal working model using an FBA model of RPOM, a rosiobacter, with DOM partitioned into glucose acetate, glutamate, and glucose 6-phosphate. But our framework is modular, and we're working to incorporate multiple more FBA models representing bacterial gills or functional groups. So our framework is also scalable and provides a pathway to simulate bacterially driven carbon cycling in Earth system models through upscaling strategies like AI ML surrogates. Thank you.
So, hi everyone, I'm Shihan Li from Texas A&M. So today I will be sharing a global scale perspective on OAE efficiency. As we have heard throughout this meeting, OAE enhances the ocean capacity to absorb CO2 by increasing seawater alkalinity. When CO2 dissolves in seawater, it undergoes a series of chemical reactions. By shifting these reactions towards greater carbonate and bicarbonate formation, OAE helps the ocean inhale more CO2. So, however, this process also drives the system toward a new chemical equilibrium in the carbonate system. Some of the initially absorbed CO2 may eventually return to the atmosphere. This is a phenomenon known as leakage. So this leakage depends on the new carbonate system state in the surface ocean, which in turn is regulated by several biogeochemical processes. So this figure, the middle figure illustrates calculated carbonate system states. The x-axis is DIC and y-axis is total alkalinity. So the contour lines shows the CO2 concentrations. The black arrows indicate some scenarios, trajectories of some different processes. As we see, photosynthesis and respiration primarily change DIC, thus the CO2 concentration is more sensitive to it. And while carbonate dissolution and precipitation, although they have it's less impact on the CO2 concentration, but it can most affect the alkalinity. Another key perspective is the carbon flux at the surface mix layer. So both organic and inorganic carbon exports out of the mix layer is a key sink from the surface ocean. However, quantifying these biogeochemical processes, how they respond to the OAE, and how they influence the OAE efficiency remains challenging. The uncertainties are twofold. So first one is we lack sufficient mechanistic understanding of these feedbacks. The second one is previous, like very traditional Earth system models are often too expensive to explore these uncertainties across many scenarios. Address this, I use Alaska model, a simplified but process-rich carbon cycle model that tracks CO2 fluxes among atmosphere, ocean, and sediments over time. It's very computationally efficient and ideal for testing a wide range of what-if scenarios. And in this project, I focus on 4 processes: photosynthesis, calcification, the vertical decay of this inorganic and organic carbon export, and the interaction between organic and inorganic carbon. So current understanding of each process will be reviewed and tested under different assumptions to evaluate their impact on OAE efficiency. With different scenarios, and our results is aim to help identify the critical knowledge gaps and also get the design of more detailed Earth system model experiments and field studies. And let me highlight one example related to calcification. Next one, please. So for this one, we compare 2 model formulations. The one for scenario 1, it assumes the calcification flux depends on the saturation state, and on the scenario 2, it links the calcification to the substrate inhibitor ratio, which is the ratio between bicarbonate and hydrogen. These 2 figures show the leakage percentage across a range of uncertainty in these parameters, eta and k. So results are shown for both calcat is a solid lines, and calcium silicate is a dashed lines, and with green lines represents for the year for the 2300. We observe from the figures that the leakage varies depending on the calcification formulation. And the other one is if the system is more responsive, that means with higher eta and higher K values, will tend to increase the leakage. And another thing we see from the figure is like silicate minerals is generally more effective than calcite in minimizing the leakage. So this is for the long-term carbon cycle, but we need to double check it with more with models of higher complexities. And from this, we see that the formulation of biogeochemical processes can strongly affect the OE efficiency. And more exciting is that when multiply uncertainty processes together and combined effect, it may amplify the uncertainty. And we are excited to see some interesting interconnection. And that's all. Thank you.
My name is Kyungmin No, and I'm the postdoc under Princeton University, collaborating with the GFDL and the Whitt people. And I'm gonna switch gear to the biology-based MCDR, the classic ion fertilization for the global perspective. So we use the GFDL SM4, which is the fully coupled carbon to climate model, which are atmosphere, land, sea ice, ocean, and each biogeochemistries are all included. So before we going on, we just defined some large-scale ion fertilization areas, which are basically on the HNLC regions and the North Pacific and Equatorial Pacific and Southern Ocean, northern and southern part. Because we divided the Southern Oceans because of the Southern Ocean northern part and the southern parts having a different water formation characteristic. For example, Southern Ocean southern parts are much on the deep convective regions, and Southern Ocean northern parts are much more related with the intermediate to moderate water. So based on these large scale areas, we designed a fertilizer experiment based on this future climate in a long term until the 2100. And we did it based on the SSP 585 C4 MIB emission driven protocols, and then we just do another red line, the keep fertilizing experiment, releasing the iron for the 110 years, and look at the durability and permanence of the additionality. We just use the stop fertilization. We did it after the 30 years of the fertilization and stop and looking at how it changes. Next, please. So we just calculated this long-term accumulated of the additionality. So when we're looking at, for example, when you're looking at the North Pacific, it saves about a 1.9 petagram carbon. So it means that for the 100 years of the fertilizations, the global ocean will take about a 1.9 petagram carbon in 100 years. And also, Equatorial Pacifics are almost zero and even a little bit of negative. And Southern Ocean northern parts are showing about a 7.7 petagram carbon and 100 year, quite significant. However, when they stop the fertilizations, they just all lose their carbon within a 30 to 40 years. So you can say the Southern Ocean northern parts are showing a weaker durability. So Southern Ocean southern part are showing keep increasing the carbon uptake, and after the stop the fertilization, they can pertain more than 50% of the additionality. So we can say the Southern Ocean southern part would be working as much more effective. So these numbers are based on the global ocean, so we can just divide it into fertilized ocean. So fertilized oceans, and we can see that on these 4 different regions that uptakes are keep increasing on this fertilized ocean. And even when you stop the fertilizations, they are not losing that much of the carbon in this fertilized ocean. However, when you're looking in the non-fertilized ocean, which is the outside of the fertilized oceans, they are keep losing their carbon and showing the large compensations. And even when they stop the fertilizations, they are keep losing the large of the carbon and leaking the CO2 to the atmosphere. So in summary, we can say that especially on Equatorial Pacific, they all lose their additional carbon by the fertilization more than 100%, and North Pacific to Southern Ocean northern parts are losing like more than 70%, and Southern Ocean southern parts are showing only the region that significant uptake on this total numbers, and they're showing the smallest number of the compensation rates. So when we're looking in the export production additionality, so North Pacific and Equatorial Pacifics are showing that additional export fluxes in fertilized ocean, and their compensations are just near the local responses to surrounding regions. However, when you're looking in Southern Oceans, their compensation signals are emerging in the tropical ocean within a different time scale. For example, Southern Ocean northern part, their signals are emerging within the 30 years, and Southern Ocean southern part, their signals are emerging like the 50 to 60 years. So because of the Southern Oceans are having a critical role for providing nutrient to the Southern tropical ocean. So this large consumption makes some nutrient robbing and nutrient trapping on the Southern Oceans and making large compensations. So our results are showing that as a MRV of the MCDR, we need to look in not only the fertilized ocean, and we need to look in the total global perspectives in a large scale application of the MCDR. So thank you for listening.
Thank you, everyone. Kristen Davis is speaking to us from Stanford University, and I will let her talk now. So Kristen, take it away.
Great, thanks, Drew. So again, I'm Kristen Davis. I'm at Stanford University in the Oceans Department. And today I'm going to talk about some work that my group has been doing to evaluate the scaling, and in this sense, I mean to make it bigger, the industry of macroalgae cultivation, seaweed farming in offshore waters. And this work was first funded by the Department of Energy's ARPA-E to really think about supporting, feasibility of a domestic macroalgae cultivation industry in the U.S. to feed biofuels, a domestic source of biofuels that doesn't require fresh water, that doesn't require nutrient additions and things like that. But since that initial project, a lot of people have gotten really excited about seaweed as a potential carbon dioxide removal strategy. And so some of the modeling tools that we were building in order to help design farms and evaluate seaweed farming also were useful for thinking about this question. So while it's not, we didn't initially sort of set out to think about sequestering carbon with seaweed, it is something that we have been thinking about since we started the project. So in order to, on the theme of this session, in order to really get at both the farming related questions and the carbon related questions, it's really been an exercise in thinking about the relevant scales and the relevant processes. And here, this kind of cheeky title, From Blades to Basins, really actually is not very far from representing the range of scales that we've been thinking about in this project. So I'm going to talk about work that represents contribution from many different groups that we've been working with as well, and I will mention them along the way. And so let's step in. Okay. There we go. So just to put the sort of in message up front, I think what we'll communicate today is, that it is feasible to sequester large amounts of carbon, it is possible to sequester large amounts of carbon by farming seaweed. Seaweed put on a lot of carbon every day. They grow very fast. That's one of the reasons that people are really interested in seaweed as a potential carbon sequestration strategy. They have a favorable carbon to nitrogen ratio, compared to phytoplankton for capturing a lot of carbon in their biomass. But really, I think some of the biggest challenges here are scaling this up to an amount that makes a dent in our global carbon cycle. And then in thinking about some of the environmental implications of sequestering that biomass, which would entail potentially, artificially sinking that biomass at depth, and what happens to those benthic environments, and what is the remineralization time on that. So there are some really important unanswered questions about not only the primary carbon pathways through seaweed growth, but also the end fate of that biomass and the long-term biogeochemical cycle. And so we're going to talk about some of the first parts of that question. How can we grow? Where can we grow? And what species would we grow to get up to the scale? And then, to think about, and then on the smallest scales, we'll talk about, how would you grow something offshore where nutrients in the surface are relatively low? So these are a range of questions that we'll talk about today. So the way that we've been approaching this primarily is through a modeling system that is attempting to represent the important processes in this problem on a range of scales. This modeling system is called the Macroalgae Cultivation Modeling System, or MACMODS for short. And in all these bubbles, you'll see some of the aims of our various projects associated with seaweed cultivation. I will talk about the ones that are in gold today. So let's start at kind of the smallest scales. We were initially using this model to evaluate different macroalgae farm designs, everything from line spacing to cultivation line depth, to orientation to the flow and stocking density of macroalgae of seaweed along lines. And so in order to think about these designs, it was important to resolve spatial scales of the farm. So from meters to kind of the size of a farm unit, which can be kind of like a kilometer, and time scales if we really want to think about turbulent transport of deep waters into the farm, all the way to a growth season, then we're thinking about timescales of seconds to a year. We'll also talk about another part of this project, which was using our model to think about farm siting. Where would you seek a permit to grow, to place your farm if you're a seaweed farmer looking to get into the business? Well, to begin to think about farm siting questions, we need to kind of go to shelf scale. And so those are spatial scales from kilometer to 100 kilometers, time scales from a day to, potentially you might be looking out, a decade into the future to kind of think about what's your yield going to look like year to year, not just, this year, but maybe next year is going to be an El Nino year, and you want to understand what your yield might look like, or over what's your yield going to be over the season to know when to plan for harvest, things like that. And then, of course, some of the questions that are relevant to some of the topics of this session is, if we want to think about the potential for seaweed farming to be one strategy for carbon dioxide removal, then we need to think, globally, and we need to be able to think about how much seaweed carbon could we generate, could we grow if we were to try to grow it, and then what would we do with that? So those are spatial scales from 1 to 10,000 kilometers and over thousands of years. Okay, so in this project, we've been fortunate to work directly with several seaweed farming companies, some in Hawaii, some in the Caribbean, and then the one that I'll talk about today the most is some work that we've been doing with Ocean Rainforest, who have a farm, a research farm off of Santa Barbara, and then also have a commercial farm that they've been working at for quite a while in the Faroe Islands. So on off of Santa Barbara, they are growing temperate brown Macrocystis pyrifera, so the giant kelp. And then in the Faroe Islands, they're growing a couple of different species, but Saccharina latissima is one that we'll be working with this coming summer in a week. Okay, so I'll start first by talking about some of the fine scale work that we've been doing. And this is some of the original work that we were doing with Ocean Rainforest, using MACMOS, which is a dynamic macroalgae growth model where the seaweed growth is limited by nutrients, light, temperature, and then hydrodynamic factors. And this schematic here is really kind of demonstrating, what you might picture in your head of a typical offshore macroalgae farm or seaweed farm, where it's too deep to grow the seaweed down at the bottom. They wouldn't get enough light, so you suspend the canopy on long lines. And in the case of Macrocystis, which is the cartoon here, they are buoyant and they would grow upwards towards the surface. Flow fields would be modified as the drag of the seaweed imposes a drag on the flow, which would drive some upward mix or some mixing in the vertical. That the farm would shade the water column below it. And would take up nutrients as the water passes through. So really, we were simulating a farm, and this was Ocean Rainforest's research farm off of Santa Barbara, really trying to understand the potential, like what would be an optimal farm design, and to try to think about yield over a season. And this work is published in, this was a paper led by a postdoc of mine, Christina Frieder, and it really sort of talks about the coupling of these three models. So a regional ocean modeling system, ROMS BEC, so physical biogeochemical model that was coupled to a large eddy simulation, which was really able to resolve flow within the canopy. So, getting down to meter scale resolution to get the largest scales of turbulence and understand the mixing within the farm. And then this dynamic macroalgae growth model, which is described here. So in this case, we're, to really simulate different farm designs and understand the implications for these, we were getting down to meter scale simulations and resolving fronds, each frond within the farm, and thinking about hydrodynamic limitations to nutrient uptake, also wave-induced erosion in losses, in biomass losses. So this is a short movie sort of looking down on this simulated farm where flow is going from left to right through the farm and slowing down along the long lines, which have some biomass. So in this case, the long lines are aligned with the flow. And one of the interesting results from this work, which was led by Yan Chow and Marcelo Chimecki, Jim McWilliams, and you can find it in JFM, is the formation of these sort of attached Langmuir type cells that would passively entrain water from beneath the farm up into the canopy through these kind of attached Langmuir type cell formations, just results of the discontinuous drag and the flow through the farm in some surface wave forcing. So this was an interesting result, and we were really eager to collect some observations on the farm to see if we could verify this. So we've been working with Ocean Rainforest at their Santa Barbara farm. This in a week we'll be headed to the Faroe Islands additionally to collect some measurements, but this is these are schematics of some of the observations. So here's a suspended schematic of the suspended farm, and we had some upstream and downstream moorings collecting hydrodynamic and biogeochemical data on the farm, certainly taking samples within the farm and outside the farm. And really the point of these are to support model development, not only to kind of validate our model to see how well we're predicting growth, but also to estimate carbon cycling within the seaweed farms. And a side benefit of this, and another direction a project we're going, is that some of what we've found is that this real-time ocean data was really useful for some of our farmers in their day-to-day operations. Just before I go on, this is my grad student, Madeline Kelm, who was leading the work in the Santa Barbara farm. So some of this work was, some of these model results were useful for different seed out dates for the Santa Barbara farm. For example, we could use many years of ROM simulations to simulate seeding out at different times and look at the implications for annual yield. We could also change the cultivation depth, and it's really a balance between a trade-off between accessing deeper nutrients versus getting more light. And then we could also play around in the model with different harvest schedules to see, integrated over the year, what would give you the most yield. So these were some of the direct, feedbacks we were able to have with our farming partners. So let's step up a scale. At the shelf scale, we've been applying MacMods to thinking about farm siting, but this is some work that I've been doing with a collaborator, Ben Sines, where the point was really to think about where would we site seaweed farms in, say, the Gulf of Mexico to mitigate nutrient pollution and the growth of low oxygen dead zones. And so certainly you all are no stranger to sort of the issues with eutrophication, nutrient fluxes from freshwater sources in that flow into the Gulf of Mexico, Mississippi River, Atchafalaya. Those nutrients cause hypereutrophication and the enhanced respiration at depth and low oxygen. And so the idea here is to think about where could we potentially cultivate seaweed that would take up those nutrients, could be harvested and used for some other use, and would this, potentially intercept some of these nutrients before they drive phytoplankton growth, and could we think about using seaweed farms in that way? So really here, the MACMODS was used to predict the growth of tropical red and brown seaweed species, Eucheuma and Saccharina, and Sargassum, and using ocean inputs from the Pisces 2 biogeochemistry model that includes nutrient inputs from freshwater sources in the Gulf. 16 years of simulations were used to create maps of macroalgae yield. And then we also overlaid those with marine spatial planning constraints from the NOAA and cost data layers. So these include things like oil and gas infrastructure, which you can see from that top plot there is quite a bit in the Gulf of Mexico, but also shipping lanes and other things. And so we were interested to see, where we might be able to intercept some of the highest levels of nutrients in the coastal regions, of course, staying out of the way of some of the, being mindful that we can't just put a farm wherever we want to put a farm. So that's the focus of this work. We are still in the sort of preliminary stage, and hopefully we'll have some exciting results on that sometime soon. So we've also been using MACMODS, was used to estimate growth for the entire US EEZ for the first chapter, macroalgae biomass chapter in the 2023 billion ton report, which is a report produced by the Department of Energy every few years to look at the development of sustainable biofuels and bioproducts. So currently there is not a sort of industrial scale production of macroalgae biomass that could be used for biofuels, but this first time it was included in the billion ton report, represents the Department of Energy's interest in seaweed biomass as a potential source of biofuels. On the largest scales, really thinking about the potential for seaweed farming to make a contribution to sequestering carbon dioxide, really we're not able, as I said before, we really were focused on kind of the first part of this question. There's some work going on by other groups that are really getting at some of the fate of that biomass if it were artificially sunk, Dave Siegel and others. But really, the first question that we got at in this work that was led by Dr. Isabella Arzeno-Soltero, who's now at UCLA, was, how much seaweed can we grow? And so really thinking about that includes thinking about not only sort of constraints on growth from ocean conditions, but also thinking about all the vital rates of the different types of seaweed. And so ISA applied our MACMODS model globally to really try to get at, if we were to try to plant, 4 groups of seaweed, temperate and tropical red and temperate and tropical brown cultivated species globally, where would they grow? How much could we grow? Just really to get at the first part of this question of how much seaweed could we grow? We can grow a lot of seaweed, but it is one of the important things to come out of this were that what we assume the nutrient fields are in the upper ocean are first order input to this question, and that we actually need to know more about the surface nutrient fields in our ocean in order to answer this question well. So the work from the outcome from this work was that indeed it was possible to harvest over a gigaton of seaweed carbon per year. So that is carbon in seaweed biomass, not sequestered carbon, but that it would require farming over 1 million square kilometers of the most productive EEZ areas. And that is a huge, huge area. And that after farming the most productive 1 million square kilometers, in order to harvest another gigaton of seaweed, you'd have to triple that area. So we have sort of reduced returns after we hit the most productive areas. So the second step here in estimating, the feasibility of this is to think about how much it would cost to grow the seaweed. And so this work was led by a grad student at the time, Dr. Julian D'Angelo, really collecting a lot of information from some of our seaweed farmers about capital costs and operations costs, and thinking about the different outcomes, not only of sinking seaweed, sequestered carbon, how much it would cost per ton of CO2, but what other things we could do with seaweed biomass, potentially make it into food products or animal feed products that could avoid carbon dioxide, greenhouse gas emissions if we were to replace the food with seaweed-based things. And this work was there were some of the models from this work were displayed on Carbon Plan's website to allow people to kind of go in and change some of the assumptions that we made in this work to toggle and have kind of interactions with the model. And so I invite you to check out that website, because I think it's still a useful way to kind of think about these things. Okay, so just to wrap up, I'll come back to this, that, from this work, we found that indeed our model suggests that it is possible to produce over a gigaton of seaweed carbon per year, but that using seaweed in place of emissions-intensive agricultural products actually seems to be more cost-effective than sinking it if you just are looking to avoid greenhouse gas emissions. And that there remain some really big questions about the implications for ocean ecosystems of sinking biomass, and that was not part of this study, but still needs to be investigated. And that's it. Thank you.
All right. Thank you so much, Kristen. That was great. We have time for, we have about 2.5 minutes for questions. So time for like a quick question or two.
How does the scaling work for half a petagram?
For half a petagram of carbon? Like how much, are you asking how much area you'd have to farm?
Yeah.
That is a good question. We have, let me see if our plot has that here. Hold on, let me get away from the chat. The area, so this is like the share of exclusive economic zone. So it's kind of in the noise here. So here, so it actually looks not entirely linear here, but it's not too far from linear. In the you could sort of guess at it to be somewhat linear in this area right here. So say about half. So 500,000.
Maybe one quick question that's in the chat is related to this. Can you comment on the scalability? The area you are talking about is greater than the sum of all ocean infrastructure globally and would require a fleet of vessels that is far behind beyond.
No doubt.
Yeah.
Yes, of course. The scaling required to achieve these numbers is one of the major issues, and it's actually a part of, it was baked into this challenge from the beginning of the RPE project. Not only were they, sort of funding innovative offshore designs, but also technologies to help scaling, like autonomous harvest vehicles and processing of seaweed, potentially at a site offshore rather than having to bring it back to port. So there was some thought to the challenge of scaling from the beginning, but it certainly doesn't erase that it is an issue. This is really on the scale of all cultivated agricultural land in the United States, just for some sense of scale. And however, if you look at expansions of seaweed cultivation in Maine and Alaska over the last few years, the growth of the industry in those waters has been exponential. It has increased quite a bit. So there is, those are not for carbon sequestration; those are for chemical products or food uses, but it does represent a huge increase. Not nearly on the order that we need, though, to achieve these numbers.
Excellent. Well, thank you so much, Kristen.
We're going to dive back into the oceans briefly. My name is Manu. I'm a faculty at Stanford in both bioengineering and also in biology and oceanography. And I want to start by just really, this session is really about scale, and I want to talk by starting about what are we up against in terms of thinking about the convergence of these sets of scales. I started my career in oceanography thinking really about the physiology of single cells and what do cells do in the ocean, and I'll show you some really surprising results that I would argue that we still know very little about what cells do out in the ocean. But then along the way, we've been building the sets of tools that we realize that we can measure certain sets of things that have not been measured before much more directly. So I'll link what we learned from the smallest scale to some of the largest scales as we go along the way. So, to just open the talk, I like to share that often enough, we all work and operate in the largest laboratory on the planet, at least. And to a certain extent, everything that we think about is impacted by living forms in this context. And this is one example of just the density of life that exists in the water column. I love this footage from David Hannon. It's a night dive. And too often when I think about this, it also gives me chills because I realize that do we even have any hope of understanding the complexity of interactions? Much of the time today, I won't talk about interactions, but you'll see that they'll play an important role at a later point. I mean, much of this is quite important. Most of you already know this, that a significant portion of the carbon is sequestered through the biological pump. And we'll kind of dive deeply into mechanistically how that might be at play and what new tools do we actually need. But this is an outstanding challenge, and many, many tools are needed. We will focus on individual organisms and individual marine snow, but there is a lot more at play that is incorporated or needs to be incorporated in the models. The view that I would like you to have for the next 20 minutes is just imagine if somebody could cut a chasm into the ocean. So we'll think quite a lot about the vertical context of the ocean. And I like to think in this context of the Grand Canyon, except if this was the Grand Canyon of the ocean, you would start seeing processes that pattern themselves along the vertical axis. And, of course, what we really care about in this perspective of the talk is really we will think quite a lot about what are the mechanistic plays in where the ocean plays a role in terms of sequestration itself. But let's just look at what do we know so far. So first of all, the very term marine snow was coined in this dive that Otis and BB went on. It's kind of remarkable. And our capabilities of visualizing the ocean have dramatically changed over time. Our imaging capabilities have changed over time. But our capacity to quantitatively measure things at the microscopic scale have not really progressed as much, I would argue. And then I'll share certain sets of tools, but it's important to remember that much of what has been described as the most stupendous snowfall on Earth started with this dive. And to show you that we know very little about the ocean, I'll ask you all to think about this puzzle as you hear this talk. These are plankton. These are organisms that are found commonly in the ocean. Many of you have seen them. These are famous drawings from Haeckel. If I was to ask you between the right and the left, which drawing is correct? And I think many of you would be pressed and challenged to think about it, because the answer is we don't know. The fact that we don't understand and know the native state of many planktonic organisms in the water column is actually by itself a challenge. These are polarized cells, so they must have an orientation associated with it. And if I was to ask you what sedimentation velocity something would fall, even a single cell or a marine snow, it gets harder and harder because these are living objects. And while we have these macroscopic scale measurements that allow us to think about the flux of the ocean, the goal of today's talk is how do we connect microscopic parameters that we can directly measure to some of these macroscopic flux terms that we're all very excited about. And then to complicate this further, we also have to remember that the living biomass actually also travels across the water column. This is almost a migration of a kilometer, happens every day. And so you can think about if something dies on the top of a surface versus dies at the bottom, behaviorally, it'll completely change the kind of sequestration flux. And it's been hard to model these sets of things because although we have the macroscopic view of this flux, I cannot tell you how long does a single cell rise in a water column. And I'm going to show you a technique that we have developed that allows us to first answer these types of questions to begin with. So, for the rest of this talk, I just want you to think about, we're only going to be talking about 1,000 meters and above. Most of the sediment traps, most of the sampling that we do is above this water column. And so we're not really talking about the deep sea, although we have started working on the deep sea with a few sets of collaborations now. And of course, everything along this water column is changing. Light is changing, temperature, pressure. So if you think about the behavioral repertoire of the organisms, these organisms are sensing many of these sets of environments. And rather than thinking about the spatial context of this, I will only, shown in this really beautiful movie from the MIT Darwin, I'm only going to be thinking about the 1D water column. And I am excited about talking to many of the colleagues to incorporate many of our models and measurements into the 2D and the spatial context. But for today's talk, we're going to ignore many of these flows and currents, and especially turbulence, because we first want to understand what does this 1D vertical context actually look like. And finally, if you needed more motivation, I just want to share this plot. This is a plot that Kale put together a couple years ago, looking at the rate at which something sinks in the ocean. This is data from marine snow captured in sediment traps, and their equivalent spherical diameter. And if some of you have thought about and think about Stokesian dynamics and sedimentation, you would realize that, this is hard. There's lots of fits that have been associated. This is data sets collected over 30 years, but we still don't really have a deeper understanding of why we see this massive variation in this connection between what appears to be the size of a particle and what the actual sinking speed is. And so for today's talk, we will show you hidden parameters that we have discovered in marine snow that can directly be visualized and measured that actually collapse this plot. Okay, so I think we're going to get there by this crazy idea, which we call ocean on a tabletop. The measurements that we will show you are through instruments that we build that are on a boat. We bring materials to the surface. Now there is work that we're doing to make these in situ measurements, but we're going to try to mimic what's happening down deep below in the water column to really understand this vertical migration. The history of this project for us actually started in Madagascar while we were studying schistosomiasis, which happens to be a parasite, and we realized that we needed a tool that could measure the vertical sedimentation and behavioral dynamics of living organisms in the water column. So we started building very tall microscopes. This is a microscope that we build that allows us to measure 5 meters of sedimentation dynamics. And you can notice as we started building these microscopes, they kept getting bigger and bigger and bigger until a moment we actually realized there is a simpler way of doing this, which is what if the two ends of this giant tube is connected? So this led to an invention which we call scale-free vertical tracking microscopy. These are two graduate students, Deepak and Hongchuan, who've been involved in this. But a very large team in the lab now have been involved in developing these instruments. And the idea is very simple. The idea is a hamster wheel for a single cell. So this is what the instrument looks like. I will refer to this as a gravity machine. You know, we're not turning off gravity. It's just a name that we have used. But the microscope on the left is essentially a traditional electronically controlled tracking microscope, while the stage of the microscope is this wheel. And so if you think about this wheel, there is mathematically two locations on this wheel, 3 o'clock and 9 o'clock, at which you can actually cause hydrodynamic levitation. This is now the instrument operating at sea. All the data I'll show you today is actually collected in the ocean. The instrument has evolved significantly. It's quite a complex instrument, and we've been building many versions of them and deploying them on boats, building a lot of autonomy in these sets of instruments. But I'll give you an example of so first of all, how does the instrument work? It's fairly simple. Say what you're looking at is a larva that's rising in the water column. We move the wheel in a tracking kind of a context such that it generates these viscous stresses along the surface that enables that a moving object can actually be in a stationary frame of reference in the lab, while in its own frame of reference, it's actually rising. Now, as I said, the ocean is complex. Many sets of parameters change along the vertical axis. So we control temperature in these systems. We control light. We now have an instrument that we can actually control directly pressure as a function of depth. But the depth is a virtual depth, which is associated with the degree of rotation in this system. So what can you actually do with this instrument? So you're seeing now, for example, just this happens to be sprinkles, candy. If you were to drop candy, but suddenly I lock in on one candy, now I'm falling in the frame of reference of that object. But now we have a capacity to actually directly measure density, directly measure sedimentation dynamics, and directly measure interactions that might occur. Say, for example, these two candy particles actually fused with each other, and we could have seen this effect. And you can see this effect in passive particles, for example. This is called kiss and run. And what's happening here is this interaction, this is dead particles just sinking, and purely using hydrodynamic interactions, you can see they attract each other. And these are sets of mechanisms that are at play when things sink passively. On the right, what you saw is a subtle movie. It is a sugar cube that's sinking. We've been studying a lot of how things disintegrate during sedimentation. And you can see the little vortices behind. These are the because of the refractive index change, the sugar cube is dissolving as it's falling. We've been applying this idea to really understand in the context of sequestration for folks that are excited about both fertilization using inorganic clays and sets of materials that are dissolved. And I think much of this is published, so I'm going to skip this part, but to tell you that there are very strict hydrodynamic limits that we apply to really make sure that we are not perturbing what we are observing. So much of the calibration data is published, and there is a limit to which that you can track an object, because many of you, if you think about hydrodynamics carefully, you could induce inertia in the wheel, and that will perturb the flow. And so we operate at this fundamental limit that you can see in this curve here, below this red curve, and there is a critical sinking dynamics which a system like this can track. So unfortunately, this tool cannot track whales migrating over because the behavioral timescales are such that you would induce inertia in those sets of systems. We also actually account for shear, so we can also correct for orientation. And what's really interesting from an engineering point of view is that this is a microscope that knows hydrodynamics. So we set these limits. And so if the object that you're tracking is moving faster than a certain time, the microscope won't move that fast, making sure that we don't induce inertia in a system. Okay, so let me just show you some data, and I'm going to switch to marine snow very quickly. But just to give you an idea that all kinds of organisms, we have currently the largest database, as far as I know, of behavioral motility, over 17 expeditions, roughly around 650 to 700 species measured across. They are all individual species that were collected in the field. you get these sets of tracks. We can actually directly, for the first time, measure dial migrations along the vertical column. We can also do this in 3D. So all of this data is actually volumetric data using light sheets. So we collect the entire hydrodynamic profile of these objects. Now, one challenge we face when you go out to sea is the sea is not flat. Doing microscopy, and especially such sensitive microscopy at sea, is hard. With our collaborator, Ben, we started using gimbals. So this is a gimbal borrowed from Hui. Thank you very much. This now allows us to actually cancel many of the tilts. So with this in play, much of the data set has been published. We are putting together new websites for people to explore these data sets. I'm not going to have time too much to talk about these data sets, but I do want to show you some fairly interesting, one interesting observation in this data set. But to just, an important thing to note is that we can literally capture a single cell traveling. This is the data set I wanted to show you. These are diatoms. And what you're watching is a single diatom sinking. And this is a phenomena that's been reported also by Gimmel's lab, and which is called blinking in diatoms. So these are not passive objects after all. Diatoms have no cilia, no flagella, and still it's able to regulate its buoyancy in 100 milliseconds. This has huge implications if I have a diatom bloom and I'm trying to understand what the flux dynamics looks like. We really have to go after individual cells and measure these sets of dynamics. So we're starting to do that with Kevin Arrigo's lab. We published, it's in the process right now, for around 50 different species of diatoms under the Arctic to really understand what the variation in density actually looks like for many of these organisms. Let me skip all of this. I think let me get to marine snow quickly. But kind of the tool, the smallest object we can track is a single coccolith. So this is sort of the limit of what we can do. We've now started tracking bacteria, but that really pushes the limit. So let me tell you, back to this object, how do we do better than this curve? How do we avoid fitting and really understand what might be the invisible degrees of freedom that might exist? So for that, we have to go back and really think about the marine snow itself. It's a dynamic ecosystem that's at play. It's being chewed by and eaten by bacteria. It has an entire ecosystem because of much of the mucus and many of the extracellular material that these things produce. And it's also extremely heterogeneous. It's made by hundreds of cells, if not more. So how do we understand the sedimentation dynamics of this object? And much of this has been talked about in the past to understand that there must exist microbial knobs to it, but we didn't have a tool to observe sedimentation dynamics of such a fragile object over long periods of time. But now, because of this tool, we partnered with several sets of partners, including Dan, Kay at Rutgers, Ben at Hui. We wrote a GCR NSF grant. And again, in these day and age, I have to say thank you to NSF, because none of this would actually ever be possible. At this point, this is our 6th expedition. I'm just going to show you data from one expedition because these data sets are quite large. We're doing sediment traps. This is all data from 80 meters, but we have now done this all the way down to 500 meters. And for the very first time, we're able to resolve sinking dynamics of individual particles. Now, we can do this during modification. So and many of you are thinking about sequestration, we can map how those sequestration dynamics would play out. But we can lock into an object, and you can see I'm showing this video just because it shows fracture dynamics that's associated. Now, I have to say we are very careful manipulating these sets of objects. We are well aware that manipulating marine snow has its own challenges. And of course, much of this data is 1 millimeter and below. The machines can handle something like 3 millimeters and below. But let's just look at this data set. What I love about this is on the left to the right, I've organized and arranged these marine snow particles as a function of size. So if you were photographing these objects, you might say that this happens to be a small particle and out here is a very large particle. But when I show you the sedimentation dynamics for this, this is the map to the right. You can see the dark, deep blue color is 200 meters per day, and the yellow color is 0 meters per day. And you notice very quickly that actually it does not correlate to the size of those objects. Some of those large objects indeed fall faster, but some of those large objects sink very, very slowly, and the other way around. And you can plot this again in that same spread that I had talked about before. And then what we realize is that there is a beautiful fluid structure interaction. So now I'm going to zoom in on this one of these data sets and just choose and show you a video. And I would like you to just appreciate if you can see something unusual. If you have taken a low Reynolds number hydrodynamics class, you should notice that there happens to be this trailing mass of fluid that's traveling right behind. We've now coined this term comet tails. Now, these are not chemical tails that people had talked about. These are physical tails of mucus. And by labeling, we've actually been able to show that this is indeed mucus. But you can now imagine that the object that's sinking is far larger than what we had assumed before. And actually, this is not a spurious phenomena. In all of our data sets, when we look, we actually find these tails. Now, these tails have a very dramatic, so first of all, we can label these tails. These tails are very stable. They're present over hours. We can measure. We can actually now watch these tails grow. And what's most important is we've now been able to build a hydrodynamic model that allows us to model the coupling between the elasticity and the viscosity of this mucus and sedimentation. Now, if you think about a heavy particle, you can eventually imagine that it's going to cause a sedimentation stress on this mucus to make this tail very long. If this tail is very long, that leads to actually a higher dissipation in the water column, and hence the sedimentation velocity slows down. And because of this, by this fluid elastic structure coupling, we've been able to now write down a new scaling law, which is described at the bottom. I don't have a time to talk through these sets of terms, but you can see that it allows us to now a new knob which is directly measured, and we can go back and look in the data itself to really be able to show what those tail sizes would look like. Now, what is most important with these sets of predictions, we can go make the measurements, we can make the predictions, and we can go back and actually check for a given mucus particle, what would be the residence time of that over a function of time. And the big implication or the result that I want to share with you, that the residence time of any marine snow particle with these comet tails almost doubles. Some of them go all the way to 0 meters per second, which implies that now they have an infinite residence time and they're going to be eaten by the bacteria, which leads to remineralization, and there will be 0 carbon sequestered. Now, this is an important sort of an implication, and in some of the perspectives, the number that we've been looking at is this might account to, if you really slow down and look at this residual time and the shift in the Martin's curve, might account to roughly between 80 to 100 gigaton. That's a very large number associated with the amount of slowdown that might exist. So just to give you this context, this is now a holotomography data sets for some of these marine snow. None of them are actually porous. There has been a lot of talk about thinking about porosity. These objects are indeed fractal, but not porous. And this is what I meant about the sets of estimates. We are working with several groups at this point to incorporate this model in a large-scale model. But one of the worrisome part is that this is actually a very large change associated with what flux would look like. In one of the perspectives that Cale wrote, the number that we all kind of agreed upon was roughly around 80 to 100 gigaton shift in the total sequestered carbon. Now, I was only able to show you, because of shortage of time, data set from one single expedition. We have done roughly around 17 expeditions so far, 5 of them completely focused on much of these, and we are starting to put together the entire data set that would be available for the community. We're starting to grow these instruments. One of the exciting parts is we are building a new instrument now that will be completely in situ and still be able to do tracking microscopy. And then the very last teasing result that I want to show you, this is work with my postdoc in the lab and also including the earlier work that I showed you with the comet tails, is with Rahul Chajwa. Now, Rahul and I and another postdoc, Ching, have also been able to actually track bacteria in these sets of scenarios. So for the first time, we can not only measure sedimentation, we potentially can also measure mineralization. And Rahul has been calling these bacterial hives, which I really like. I'm just going to show you what these hives look like. I'm now tracking these bacteria. This gives us a sense of thinking about who's arriving on these particles. What does the disintegration of these sets of objects actually look like? I know I'm almost out of time, so I'm just going to close here by just saying it's been an incredible journey, I would say, in terms of working with many groups across in thinking about these sets of ideas. And one thing that I am really excited about is being able to take these sets of models and measurements to scale and to be able to build them in large scale models that really now account for productivity in the sets of oceans. But it's very important to think about how cellular behavior and microbial processes for the first time could actually be accounted and directly incorporated in some of our Earth scale models. If any of you are excited, I would be very happy to chat. And also, if there is time, I can take questions.
Thanks, Manu. Let's see if can everyone hear me? I don't know if this is Dan Whitt, but I can't go on video. That was an awesome talk. We are a couple minutes behind. Thank you for doing exactly 25 minutes. What I would suggest is maybe we can, you can engage in a chat discussion on the chat, and then we can go on, switch over to Melissa. Melissa Omand is a professor at University of Rhode Island.
Thanks, everybody. Thanks, Manu. That was super exciting, and leads into some of the same kinds of things that I've been thinking about in the ocean as well, but in terms of developing in situ measurements to be able to look at more of the details of sinking particles. So I'm going to be talking about low-cost technology to measure sinking fluxes and giving some examples from coastal margins to the deep ocean. And this is thanks to collaborations from my very wonderful colleagues, some of whom are listed here. So just to kind of summarize one of the main messages that I wanted to communicate for today, I mean, I think there's a need for distributed measurements within our science community, and also importantly, an expansion of the tools that make these instruments available at different price points that will enable a much more broad participation across more groups. And this might include the private sector, industry, as well as sort of education level and developing countries that are working on building up their own science capacities. And there are lots and lots of low-cost tools that we can now use for this. Microcontrollers, computer chips, very inexpensive cameras, and lots of sensors that the prices are coming down for, as well as some tools for remote data telemetry and localization. And so, as I mentioned, I'll be showing some different applications that span some of these different kinds of environments and sort of applications. The first one I'm going to talk about is a float-attached snow cam. And this is data from the Exports North Atlantic cruise. The next is data from Monterey Bay, looking at some results from Minion floats. And the third is some surface tethered and bottom landing flux camera examples. And this is work that actually I'm going to be doing coming up this summer in coastal Ghana. So a snow cam is basically a very simple instrument. It's an upward looking camera that has can you guys see my mouse?
Yes, just move it a little bit slowly. It doesn't
Okay. All right, so it's an upward-looking camera inside a housing. This one's rated to about 1,000 meters. It has internal recording and batteries, and it basically takes photographs of marine snow particles that land on this upward image face. There's an LED ring that sits around the image surface that allows the area to be illuminated when we're in sort of darker parts of the twilight zone. And it also has some ancillary sensors like pressure, temperature, and ways for communicating and turning it on and off. And so I wanted to show an example of SnowCam image that we collected last summer. This is from an instrument that we call the SNOWCam Plus because of its additional capability to act as a sediment trap controller. And you can see the accumulation of particles, lots of different types of particles, and also some of the movement that's happening. This was on a SNOWCam attached to a Lagrangian float. So it's in a drift mode, but there's still a fair bit of motion between the float and the water environment. And because this is open face, you do see a little bit of movement associated with, I think, that water motion, as well as potentially some other kinds of animal interactions. And this is a really improved image relative to some of our first images from the exports North Atlantic, which can be shown on the left-hand side there. So moving back to that data set from the North Atlantic, though, because it's the sort of oldest one, the most mature one that we have, and the one that we've sort of processed to the point of estimating carbon flux. This is a trajectory shown of the Lagrangian float, which is in pink, and a sea glider, which is shown in white. And so the float was drifting around an anticyclonic eddy, and the sea glider was following after it. And so we are getting profiles continuously in this Lagrangian frame. And so now what we're looking at is a time series of the glider data. The top panel is density, and we have the mixed layer depth shown in the black dashed line, and this spans about close to 2 months of deployment. And so for the first month, the gliders were out on their own. For the second month, the ships joined them, and the Lagrangian float was deployed. It's shown in the black line, the squiggly black line there in the top. And at that point is when the sea glider started to follow it. The bottom panel here is optical backscatter measured by the sea glider, and it gives a proxy for the amount of organic material or particles and cells in the water. Now that can be processed into two sort of categories, a baseline and a spike product. And so I'm showing the baseline product on the top and the spike product on the bottom. The spikes is basically just the anomalies relative to a median filter. And so it's providing an estimate of the abundance of large aggregates, we think, that create spikes in the backscatter. And what really stands out here beautifully is that we see a decrease in the total surface concentration in the baseline, which we can attribute to some of the small, sort of more likely phytoplankton particles. And then around May 15th or so, we see an increase in the number of spikes or large particles in the surface. And a little after May 16th, we see this plume of spikes descending in the water column at a speed of about 50 meters per day. And so this is really a beautiful example of sort of a succession of surface, phytoplankton communities that something has changed, and those communities are now, beginning to form particles, aggregates here, and sinking out of the water column. But we don't get any more information about these spikes using the biooptics available on the glider. And so they don't reveal particle identity. And that's where I think there's an opportunity for a tool like a SNOWCam to come in. So the top panel here, we had our SNOWCam attached to the Lagrangian float. And the top panels here show some of the image processing that we had to undergo. So it was because it wasn't too deep, it was fairly clear. We're still getting a lot of background light. We had to do a background subtraction, do some binarization to be able to detect the particles. And then we processed the images from there. And so the middle panel is the Lagrangian float time series, and each of the purple squares are where we took a photograph. So unfortunately, I was a little conservative on battery life here. I was nervous that we were going to run out, so I only took about 2 or 3 photos for every drift. So the Lagrangian float did about 12-hour drifts and then would come to the surface. And each time it came to the surface, the snow cam basically rinses off and then it comes back down and resets. So the bottom panel here is an example of 3 consecutive images where we see even in that 12-hour time period or less here, I think it's, we have quite an accumulation of fluffy aggregates. And we can process those images and compare them to some other estimates of carbon flux. So on the same platform, we also had what's called an optical sediment trap, which is a transmissometer-based tool for estimating carbon flux. And we also had lots of in situ direct measurements of flux from our sediment traps attached to some neutrally buoyant sediment traps and also a surface tethered trap. And so using these, we can construct an estimate of the flux based on attenuants or essentially based on area coverage from the images. And they agree really nicely between the OST or pretty nicely between the OST and the SNOWCam product. And they give us information about what types of particles are there, what size they are, what their particle identities are. On the bottom panel, I'm just showing some of the ratios between and the distribution between the large and small particles. And there's lots more that I could get into here, but I'm going to move on for the sake of time. Basically, the main message that I want to convey here is that I think these cameras are a promising, very low-tech tool, and they're really best used in amongst a suite of other measurements to verify what they're measuring and also provide some important context. Next, I'm going to move on to minion floats. And these are basically snow cams with some extra bells and whistles and sort of deployed in a standalone way. So we can hopefully produce them and release them in larger numbers than we would be able to if every snow cam needed to be attached to a pretty expensive Lagrangian float. So minions are about 22 inches long. They're made out of glass. They have one end that's rounded top here that has our iridium, GPS antennas, and strobe. And then they have some circuit boards, primarily a Raspberry Pi Zero and some other custom boards that we've built for controlling the Pi, sleep cycling, and doing some of our ancillary stuff, as well as what we call the MinSat board, which does the Iridium and GPS communication. Below that, there's a battery pack, and at the end cap is where we have the sensors and the imaging surface. So minions are made as inexpensively as possible and as small as possible so that we hope we can deploy them in sort of large numbers in fleets that can distribute through the water column to give us measurements of the vertical attenuation of particle flux, as well as some broader spatial coverage. And so they don't profile, they have no ballast control, which is part of how we keep them inexpensive. Instead, they're pre-ballasted and then trimmed on board after doing a CTD cast to just the right density to allow them to land on target isopycnals. And then they drift for a period of time, taking photographs, and at the end of their mission, they drop a weight that makes them positively buoyant. And now the battery side becomes the heavy side, and the minion will flip over and have its communication suite facing upwards and allow it to transmit data and strobe to be collected. And so here's some videos of a minion deployment, the sort of flipping motion underwater, and drift that we recorded from the ROV Ventana at Mbari. And so we were able to release a minion from the ROV and then follow it for a while. And I think we were at a depth of a few hundred meters here. And you can see some of the curious animals that come to check this out. It was fun to see this from the ROV, and we've also seen evidence of this in some of our photos. So here's a example of minion time series. And this is one, I believe this is one where we have like that siphonophore, we think, in the background that's sort of sweeping some of the particles around. And then we have a little crab that actually landed on the image surface and basically crawled around and swept up particles. And this was, for context, the time in between each of these images is about 10 minutes. So the crab was sitting on the surface there for quite some time. And Colleen actually provided some image of an example of one of these little pelagic crabs with their sort of, it looks like their belly is full of marine snow. So I think that this is just sort of a vicarious example of how we built a tool for one application, and we have the chance to discover some other sort of novel things along the way. But it does make our image processing a little bit more challenging. And so our lab has worked really hard to not only be able to identify and classify particles, but also do particle tracking over time and detect events. And so what I'm showing here is an example of a few different approaches we've taken for detecting events based on the correlations of particles before and after subsequent images. And so another challenge, in addition to, the fact that these particles are, have quite a bit of movement and other things happening, we also can get, we can also see some issues with overcrowding. We saw that with our first image from the SNOWCam that I showed. And I think this is actually a fairly tractable problem. There are a few ways forward. One of them is just that we've tried to basically do a sort of Monte Carlo style modeling of particles with a, with a known, or simulation with a known arrival rate, and look at how overlapping of different particles affects what we measure as the size of the particle and the overall area. And so we see in our data sets often a rolling off of the total area, or the number of particles that we detect. And so that's what's shown in the bottom on the left. And we can actually correct for that based on some of the modeling that we can do to be able to to be able to back out what we think is sort of a real change versus issue with overcrowding. And overall, I think, a best solution here would be to, a priori have an idea of the environment that you're deploying in and make a decision about the deployment duration, or incorporate a mechanical wiper. So we have some efforts underway to do that. Another sort of fun a new project with the minions is our progress on a side-looking camera. So in addition to having an upward-looking camera on the end cap, we've also now incorporated a sideways-looking camera shown here. And we mounted this to the side of a profiling float from UMaine. And what I'm showing here is actually an example from a laboratory setting, not from the field. From our field, unfortunately, we had very, very dark images and it was fairly sparse. So we're still, we do get tracking of particles, but I wasn't quite ready to have that data process to be able to share today. But I think it's very promising in terms of, you know, getting to a point where we can estimate sinking fluxes from floats like this. Let's see, I think that one thing that has really, this work has highlighted for us is, the importance of understanding what the float water motion is. And so how Lagrangian are the minions? Here we have a time series of pressure from 4 different minions. So they each have names Ika, Ame, Buta, and Shi. And they've been deployed each for about 18 hours. So you can see where they were deployed, where they pop up. And these were deployed on slightly different days. So I'm putting them all in hours since deployment. But Buta and Shi were deployed at the same time. And so you can see some of the correlation between the pressure for both of those. Whereas Ika here has a really large excursion associated with the internal tide. Now, what's really interesting is that you see all these different wiggles in the pressure record, but you don't see any corresponding correlation between the temperature and the pressure at those high frequencies. And so we can look really, really closely at our temperature record and look for any evidence of the minion sort of traversing a vertical temperature gradient here, and we don't see that, which is really suggestive of the minion following internal wave oscillations with these high frequencies. We do find that we see some correlations on the low frequency, so especially for Ika here, that one has a little bit of an inverse correlation in pressure, suggesting that in this case the minion wasn't completely keeping up with the internal tide oscillations. But this is all really kind of exciting and interesting results that sort of point to how closely these floats are following the water. And we can go a little bit further because we have a CTD profile from the deployment. And so we can take advantage of the gradient in temperature to be able to estimate the predicted excursion of the minion relative to the mean isopycnal or mean isotherm that it's oscillating around or near. And so what's shown in black on the left-hand side is the actual depth of these 2 minions, Buta and Shi. And then in pink is the predicted displacement of the minion relative to an isotherm. So that's really telling you how much of an actual displacement the minion is experiencing relative to being fully Lagrangian. We've also had some really interesting results and good progress toward understanding the behavior, the Lagrangianness of the minion using numerical modeling. And so because of our very careful ballasting process, we know an incredible amount about the minion's mechanical properties, its mass, its density, its compressibility, its thermal expansion. We can do a really pretty good job of parameterizing its drag. And this allows us to build a numerical prediction of how closely the minion might follow the water under different sort of environments. And so what I'm showing on the top here is the predicted trajectory of the minion, shown in red, relative to its target or center isopycnal, shown in black. And you can see here it's following it very, very closely. Whereas if the minion landed slightly deeper in this water column, in a place where we have much weaker stratification, we can see that we would then expect some departures from the isopycnal. And so the relative float water motion would be greater in this very unstratified region than it would be in the stratified region. And so I think this helps us a lot because it gives us the chance to really decide where are going to be the best places to go and use the minions to be able to estimate sinking rate with the side-looking camera. And so, in summary, I think the minions really offer an unprecedented and, very Lagrangian insight. And this may allow us to estimate sinking rates. I didn't talk about this in this talk, but it also will allow us to measure oxygen respiration in a Lagrangian frame with our presense oxygen optode. And it allows us to also look at the visual decay of particles, because as they stay on the surface for a period of time, we can actually look at how they're changing. And so in my last section, I wanted to just touch upon upcoming work in terms of using snow cams to look at carbon flux in coastal environments where Lagrangian floats or minions might not be well suited. And so we actually went out, we're going to be doing some sediment trap deployments in Ghana this summer. And on the right-hand side, you can see a sort of cartoon of a very simple sediment trap array with tubes at 50 meters and a snow cam at 50 meters that will allow us a single depth collection. We went out last January and did a pilot cruise, just sort of shaking down the Ghanaian Navy vessel that they're generously allowing us to sort of retrofit into a research vessel temporarily for our cruises. And I wanted to sort of share that I think the SnowCam really works very, very well in these very low-tech applications too. So this, if you just add a sediment tube above the SnowCam and have it on a surface tethered trap, it actually works remarkably well and has very little particle fluid sort of motion. And I also think that we can have a circumstance where we have a benthic lander or a bottom landing application as well. So here you can see the accumulation of particles from the surface tethered trap. How am I doing on time?
We're about 1 minute. We're over time, so if you could wrap up as
Okay.
quick as possible. Sorry to bother.
Yeah. Yeah, I just wanted to, here's my slide on tracking the particles over time. This is for the sort of changes in particles. And I think my final slides are just that I think that truly bridging these scales, it requires a fleet of different platforms. So we want to fit these imaging floats into the existing array of satellite and glider and BGC Argo observations that sort of provide such a rich context, and that we're really adding to a family of observational or imaging platforms here. And so some challenges for open source work is that, it really is important to support users as hardware goes obsolete, as we get updates, but we have endless opportunities for modifications and new applications. And I think that in terms of low cost, our challenges are just being able to balance data quality and ensure that tools are used alongside verified methods. And our opportunity is just broader access and more persistent data and a way forward when funding is limited. And so thank you. Sorry for going a little over time.
Thanks, Melissa. That was fantastic. I'm going to recommend again that we punt the questions to the chat. We will have a panel discussion, hopefully with all the keynote speakers at 3:45 Eastern, so we can also continue the discussion then. Right now, it's time for the lightning talks.
These are results looking at marine snow dynamics during the demise of the North Atlantic spring bloom using in situ particle imagery from the UVP5. I was looking through our collection of pictures and I, for some reason, couldn't find any of the really nasty wave photos. I guess nobody wanted to take. Okay, what's shown here, these are vertical profiles of the spectrum of the particle sizes done in particle volume. So it's the particle size spectrum multiplied by the size of each particle. We're doing it this way so it shows more of the things that are going on. So as Melissa said, we came out and we could sort of see we see an increase in these large particles. The black line. Oh, I don't have the black line is where the daily mean mixed layer is. These are selected days. The storms are between the storm symbols there are showing when we saw storms. We can see that the mixed layer particles become more numerous as the cruise goes on from like dates like May 12th up to the 19th or so, and they become larger. You have more of them coming up to the surface. And this is contrary to trends in POC, trends in NPP, trends in biogenic silica or whatever, which showed a decrease in these surfaces. So initially, the particle volumes at depth below 100 meters or so were there were very few particles, and eventually we see this plume starting at about on the 14th and going through there. This plume of particles that are about 3 millimeters in size, and that are sinking into the interior. The big clue here that I want to talk mostly about is that if you look at the population of particles in that top kind of 20 meters, you can see that within the mixed layer, the particles numbers are the particle sizes and numbers of large particles are decreasing as we approach the surface. So there's a hint there as to what's going on. I'm going to talk mostly about that. Okay, what's shown here is a time series of all the daily averages on the top. In the blue is shown what the total particle volume is. And particle volumes initially are quite small, but after about the 12th of May or so, in that second set of samplings, we can see that the particle volumes double in just a couple days. And a storm comes and they kind of break down again, and then they grow, and then they again a storm comes and washes it out, and so we can estimate the kinetic energy dissipation rate of what is the scale of turbulence in the mixed layer using the air-sea interaction processes, and we see a strong relationship between calm periods where the particles grow and rough periods where the particles seem to get destroyed. The panel in the middle shows the spectrum now for the upper 50 meters averaged, and it shows both so you can sort of see that, especially in the second and third, you can sort of see that the numbers of large particles are growing. The red line is from laboratory experiments that Alice Aldridge did many years ago, but that supports sort of this upper bound of as to where we see particles, where we see the largest particles. Also shown on this are the coagulation rates of very large marine snow particles. So these are marine snow greater than 5 millimeters. We can see that this coagulation rates follow this general pattern. So on the bottom panel, we estimate also the fragmentation to coagulation rates. We're looking at the ratio, and when the turbulence is high, we have more fragmentation. When the turbulence is low, we have less fragmentation. The other thing to talk about then is that these particles are incredibly porous, as Manu and Melissa showed earlier. They are the porosities we've estimated from a bunch of other techniques, but and it shows that these storms had proximate controls in the upper layers marine snow characteristics, both their size and their porosity, and in some sense they set them up, and it had a lot to do with the timing of when that plume of sinking particles showed up, and that this fractal nature needs to be shown for any analysis. Next slide. Okay, so
please wrap up quickly if you can to keep us on schedule.
This is it. There's a preprint with a QR code. There's lots to talk about. The sequence of storms transported marine snow to beneath the mixed layer where they could eventually sink. It allowed them to grow without having the storms break them up. We have also evidence of biological processes, both taking large particles and making small particles out of them, and it's likely due to interactions with zooplankton. And the results overall illustrate the complex interplay of among the physical and biological processes regulating the dynamics of marine snow and sinking fluxes during our experiment. The preprints available for anybody. I'd love comments. Thank you so much.
Hi, I'm Stephanie O'Daly. I'm a postdoc at University of Washington in Seacos, and I'm going to be talking about an application of unsupervised clustering to classify 5 million marine particle images. Thank you. So, and one more advance. So we worked with the US Ghost Ship program, and we deployed an underwater vision profiler, which is actually the same instrument that Dave was just presenting data on from the particle size spectrum. And I'm going to be talking about the image data that this instrument collects. And we worked with the Ghost Ship program and deployed this on 300 full depth profiles. And then we had 5 million images to sort and another advance. So the problem with this kind of data is that with images of marine particles, usually about 90%, over 90% of images are detritus, and it's very difficult to classify these images because they're extremely diverse in how they look, and it's difficult to create training sets that accurately exemplify specific categories within them. So it creates a problem for how to analyze this kind of data. So we use the morphocluster technique. This is a deep learning algorithm that uses clustering. So you put in the image with no metadata associated with it, and it creates these specific clusters of individual images that look similar to each other, and it presents those clusters to the person validating it. And you either accept the validation or you reject the validation. And if you accept it, then you start to expand the boundary in the growing phase of trying to encompass all the types of particles that are within that category. And then you send it back into a clustering phase with everything that hasn't been classified into a category and repeat that process. And then once you get a good number of particles classified, then you create a hierarchical arrangement of the different categories and you name those categories. So this is really helpful because you don't go in with any preset notions about what you're going to find in these data. It's completely data driven and you don't need to predefine the categories. So we did this and we reached 91% classification using the morphocluster technique, which was 46 detrital categories and 62 living categories. Advance. And all of those can be encompassed in these 12 super categories. And this is just the overall distribution of how many images were in each of those. Advance. And if we look at the one of type feces, we saw that 9% of all images that we saw were feces. And so this is kind of exciting because I think a lot of times we tend to assume that large particles are aggregates. And so a substantial amount of these particles were feces. So I'm going to spend the rest of this talk just going through those results. The feces were broken down into 16 different categories. And I just chose an example image from each of those categories and showed how the distribution of those different types of images. And this type of high resolution classification is pretty much only possible using this kind of deep learning algorithm because it would be very difficult for an individual to keep track of all these different categories and how they're different from one another in order to sort these. And also it would be difficult for us to know ahead of time to specifically look for these types and advance. And then it also, we can see that some of these are linked with specific taxa. And then so there's copepods, and then if you click one more time, and then again, there's euphausids and salt pellets. So those are things that we can kind of more clearly link with specific biology, but some of those are also more of just degraded particle fecal pellets, and then advance. And yeah, so here's just a little taste of like what the full profiles look like of just all the fecal pellets in these 4 different cruises and different ocean basins. And if you click advance, there's we can see like there's high abundances at the surface and at the coast, and then there's areas where those patterns are different. And so that leads us to be able to investigate why we're seeing high abundances in different areas and if they match with the location of zooplankton or not. Okay, that's the end of my slides. Thank you so much.
Thank you. Yeah, hi everyone. Thanks for having me today. My name is Mona and I'm a postdoc here at Dreux. So what if there are system models we use to protect our coastal ecosystems are missing parts of the story? I will show you why we need high resolution predictions of bottom water aragonite and how we can get these. So the reason why we need high resolution data of aragonite is ocean acidification, and due to increasing CO2 concentrations, which not only decrease the pH visible in light blue on the first figure, but they also reduces the availability of free carbonate ions, which are needed by calcifying organisms such as mussels and plankton to maintain and build up their skeletons. If we want to identify acidification trends under climate change in Earth system model predictions, we have the problem that these are mainly in coarse or low resolution, since high resolution predictions have high computational costs. These low resolutions mainly vary around 1 × 1 degree grid cells, like on figure B, and therefore miss small scale signals and limit the evaluation of local processes. One low-cost solution to get high-resolution bottom water data and to estimate and locate the effect of ocean acidification is to use convolutional neural networks, CNN. And these are a deep learning approach that capture and preserve complex spatial patterns while the resolution is increased intelligently. And the CNN approach is divided in 2 steps. The first step is setting up a CNN trainer, which is based on daily high and low resolution data of GLORIS and matching carbonate computations. And the second step is applying this trainer then to monthly low resolution Earth system model outputs. CNN uses mainly gridded input data, often for image recognition, like on figure A. And as mentioned before, we use the daily low and high resolution GLORIS data as reference data pairs for the training, and then apply the CNN to monthly low resolution Earth system model data. The CNN then applies the first convolutional filter, which recognizes specific features and patterns and creates feature maps out of them. And additional convolutional filter then combine the simple features in more complex features. And at the end, the fully connected layer let the CNN produce the image, which is in our case, the high resolution Earth system model data, which is visible on figure D. And as we can see there, the CNN did a great job, and the spatial features from the high resolution GLORIS, like for example, the region around the Gulf of Maine or the Georges Bank area, are now nicely visible in the downscaled Earth system model predictions, so that these small scale characteristics can be investigated. In order to identify acidification hotspots and check the CNN predictions, we compared them to GLORIS. And we see that especially in the freshwater influenced regions along the coastline, we have higher errors visible in yellow in figure A, and see that in these regions, the CNN predictions underestimated GLORIS visible in blue in figure B. We also compared the CNN predictions on figure C, which in the darker colors, in more detail at different locations, not only to GLORIS, which is the black line, but also to a simple interpolation method, which are the light colored lines. And the temporal patterns show how good the predictions overlap, but we also see that the predictions work differently well depending on the location. So we are still investigating this, and one task is to identify possible drivers for the differences between the 3 predictions. And we are also in the process of doing all the same with a half-degree Earth system model input layer for comparison. So since the overall goal is to identify acidification hotspots also in future climate predictions under different climate scenarios, we see on the right plots a comparison of aragonite from GLORIS in December 15 and the downscale predictions for aragonite in December 2100 under the business as usual scenario in figure E. And this clearly shows a drastic acidification in the entire Northwest Atlantic with aragonite under saturation in most parts of the region. And the delta between the 2 in figure F shows again that the highest change in magnitude, or like the strongest acidification, would occur on the shelf along the highly dynamic mid-Atlantic bight. Yeah, with this, I thank you for your attention and summarize that CNN downscaling can help us to identifying acidification risks at local small scales in current and future predictions at low computational costs.
Great. My name is Bro Johnson. I'm a research faculty at the University of New Hampshire, and I'm going to talk a bit about timescales and how to measure the variability in timescales in marine ecosystems, especially using chlorophyll. And part of this work is coming from some really nice old papers by Ken Biesler from '98 and around there, where he was talking about how a lot of the carbon export production might be driven by differences in variability in the different phytoplankton ecosystems. Next, please. This is an example where we started our thinking. Here is a time series from OCCCI, which is a chlorophyll satellite product just somewhere in the North Atlantic. And you can see daily data. We can see that there is no real trend. There's a lot of seasonality in the data. This looks great. And since we're really good at picking up structures, we can see that there's seasonality. But we wanted to know, what is the dominating timescale? Is it fast or slow? And the problem is that satellite data is extremely sparse. So here's an example of where we colored every day where there's no data as red. So you can see that most of the days we actually don't have any information. And almost all statistical methods for looking at spectral analysis or variability don't really like nans, or really don't like no values. So we developed a new method where we take so basically what we do is we run the standard deviation, the running standard deviation over the time series. So in top you have 3 different time series, or you have one time series, and you just have a window of a running standard deviation, and you calculate the standard deviation in every window, and then take the average, and then you calculate this curve at the bottom that shows how the variability increases when the window size increases. And at the end, you can see that this saturates. So we basically see the entire variability in the data. And the nice thing with this is that when we have different time scales of variability in the signal in the top, this curve changes in slope. So a slow changing curve gives a shallow slope, and a fast changing slope gives a steep slope. However, the sparsity doesn't matter. At the bottom, you can see that if we removed up to 80% of the data, the slope looks exactly the same. So that means that with this method, we can pick up what the dominating or the saturating variability in this time series is. And this is just an example of 2 areas where we can see in green chlorophyll and in blue sea surface temperature in the Pacific. So it's 160 west and 18 and 35 north. And you can see how these scales, these slopes changes very, very nicely with how fast the variability is in the different timescales, in the different time series. What we did after this is to find a way of defining the slope of the curve and plot it on the global scale. And next, please. We get these figures. So at the top, I apologize for the ugliness. That's me converting it to Google. But at the top, you can see sea surface temperature and the dominating time scale. And at the bottom left, you can see chlorophyll dominating time scale. You can see that's a tremendous amount of wealth of information here. You can see that certain areas have very, very short time scales, not in the gyre, but just north of the gyre. You can see the coastal areas have very, very short time scales. And then you have these seasonal scales of variability in just above the north of the ACC. So you can start to see how these structures look. And then when we zoom in to much closer areas, we can see even more structures. So you can see to the right how we have very, very complex tidal structures in the Long Island Sound, and that around Woods Hole, you have a very short time scale. And then you can see where we start to pick out the seasonality in the oligotrophic part of the North Atlantic. So we think there's tons of information using this method in understanding how different scales affect the ecosystem, and how then at the next step, how can this affect export production and zooplankton biogeography, et cetera, et cetera. Thank you so much.
Good, thank you. Hi, everyone, and thank you to OCB for the chance to share a little science.
I'm going to be speaking today about the Marine Biodiversity Observation Network node in the Northern California Current. We're integrating technology, theory, and machine learning to track biodiversity and ecosystem functioning, scaling from the cell to the seascape. We've built this as a collaboration between academic and agency scientists. And I put a QR code up there because fingers crossed, we're maybe recruiting a postdoc, federal landscape withstanding. So please reach out if you're interested. All right, so a large scale circulation and local intermittent upwelling drive high productivity and high secondary productivity in the Northern California Current. This region is changing. Marine heat waves, changing wind patterns, hypoxia are reshaping the community structure and biogeochemical cycling. On the bottom here, you can see that upwelling favorable winds may be intensifying, and this may enhance nutrient delivery, but it may also decouple or displace trophic interactions. And this affects carbon cycling and fisheries productivity. If you could see on the left, the latter has been measured historically through NOAA fisheries surveys. However, it's really, really challenging to redesign these surveys and or expand net-based studies to greater spatiotemporal scales or taxa. So our MBON node builds on NOAA's legacy observations by adding taxonomic depth, higher frequency imaging, and scalable AI tools to address these challenges. So we embed our in situ biological data within dynamic seascapes shown on the left. It's a global classification of satellite modeled oceanographic data. And each class, so each color that you see, represents a different biophysical conditions, as well as the phytoplankton functional response to those conditions. And so within those, we collect high resolution observations through imaging flow cytometry, hyperspectral IOPs, a towed imager for zooplankton and ichthyoplankton, as well as periodically sampling for environmental DNA. So as you can see on the right too, we also conduct targeted experiments to probe trophic coupling. These include dilution experiments and carbon pathways through oxygen tracer measurements. So we use this to test ecological mechanisms. For example, linking diatom dominance shown on the bottom to food quality and export efficiency, and to develop these indicators that are meaningful across disciplines, fisheries and carbon cycle alike. So our plankton images now number in the tens of millions. So I think I underestimated that. It might be up on the order of 70 million. And so to scale their use, we've built AI pipelines that combine supervised and unsupervised clustering, morphometric feature extraction as well. So these models are being integrated into an open source and web-friendly pipeline to support diverse image types and architectures. So you can see the current pipeline on the right. Our phytoplankton pipeline is in the middle, and then the merge pipeline will be on the right, using all open source web interface, as well as YOLO as the CNN for classification. So we extract size and shape metrics to estimate biomass and functional traits. And next steps include merging environmental metadata and optical properties to allow trait-based mapping of plankton across the seascapes, and supporting algorithm development, biogeography, and inference of carbon flux processes like growth, grazing, and net community metabolism. So go ahead and, we can advance a couple, and you can see some of the players. It's just a little plankton porn. So here we go. These are the heterotrophs in our area and the autotrophs, primarily diatoms in this region. Okay, next slide. All right, so here's how this works in practice. In spring, remote sensing shows cross-shelf gradients in chlorophyll A, and community structure may vary with chlorophyll A. But if you want to advance one, you can see cross-shelf variation in here, and then one more. It also may vary by the physics forcing a bloom. So by embedding the multiple cruise observations within seascapes, we can account for those biophysical nuances and see a few things. That community composition is different across seascapes on the left. You can see across multiple seascapes that diatom richness as well as phytoplankton particle size are different across these seascapes. And then we can exploit these differences to optimize remote sensing algorithms. And in the bottom here, we've modified a pigment-based diatom algorithm to include the spectral slope of backscatter from our in situ IOP package. And this boosts prediction of diatom relative biovolume, a proxy again for the food quality and export potential. And so seascapes here constrain and contextualize these models. All right, next slide. And I think that's my last. And so we didn't have time to talk about the broader scale vision in terms of the growth, grazing, and net community metabolism. But I just want to end with the Northern California current is shifting. Our tools, our AI tools, help us detect, interpret, and scale these shifts across trophic levels, ocean conditions, and carbon pathways. Seascapes are operational. The QR code there is for NOAA Coast Watch, and it has uncertainty and novelty layers built in, and they offer a scalable lens to detect change in ecosystem structure and function. And so we hope that together these tools strike a balance between mechanistic understanding and data-driven inference, allowing us to rapidly assess change while remaining grounded in ecological theory. And I'll stop there and thank you.
All right, that was awesome. I want to thank all the lightning talk presenters for, giving us a really, really great mixture of topics and staying more or less on time. We only lost like 5 minutes there. So we can keep chatting in the chat. And now we're going to transition to Naomi Levine, who's an ex-plenary speaker, professor from University of Southern California, talking about breaking open the microbial black box to improve our understanding of biogeochemical cycling.
Thank you so much. And thank you for a really fantastic session today. I really enjoyed all of the talks. So I was really excited to be invited to speak in this session because bridging scales is really the primary focus of my research. So like most of biogeochemists, the science questions that I ask are at large scales. What drives large-scale patterns in the carbon cycle and variations? As I like to illustrate using this, can you see my mouse? I'm doing this. Maybe.
Yeah, we can see the mouse there, but it's hard if you go slow.
Okay, I'll go slow. So I think this ocean color image from NASA really nicely illustrates how dynamic our ocean carbon system is. And so I want to understand that system and how it will change as the climate warms, as the extreme events become more frequent. But while the science questions that I ask are at the scale of the globe, I really believe that in order to answer those questions, we need to understand the underlying microbial dynamics that occur at the scale of individual microbes, which are driving the carbon cycling, which requires zooming in down to that microscale to uncover underlying principles that are governing the dynamics and then scale back up again. So at the really most fundamental level, what I'm interested in understanding is this simple equation, the rate at which inorganic nutrients, carbon, nitrogen, phosphorus, and others are converted to organic matter and back again, and what drives variability in those rates, which are represented by these two purple arrows. Specifically, how are these rates impacted by microbial dynamics, such as microbe interactions, microbe-environment interactions, microbial community composition, or behavior? In other words, why does it matter who is there and what they are doing? For understanding these rates of conversion. So in my research, again, we're zooming into the microscale to develop a mechanistic understanding of what's driving variable rates of production and degradation of organic matter. But then we try to scale back up, going from cells to ecosystems to understand the large-scale biogeochemical impacts. So to do this, we're mainly a computational group. We use a range of different numerical models that allow us to do this scaling. In addition, a really big challenge that we think a lot about is how we can connect the current state-of-the-art microbial observations, such as the omics measurements, with these large-scale biogeochemical models. It's a huge challenge of scale, and right now the models are not formulated in a way that facilitates this engagement and interaction. So today, I'm going to give you just a few vignettes of some current ongoing work in my group that we're doing to bridge these scales. And because it's such a short talk, I'm not going to really be able to provide a huge amount of detail on these studies, but I'm really hoping it will spark questions and discussion either today or offline. Feel free to reach out to me. So the first example I'd like to show you is how we're using cellular level decisions to inform large-scale biogeochemical models, specifically for dissolved organic matter or DOM cycling at the global scale. So this is work that's being led by 2 graduate students in my group, Ryan Reynolds and Anna Weiss, in collaboration with Cameron Thrash here at USC. And we asked a really simple question: could we try to understand and define marine metabolic niches, microbial metabolic niches, by building and interrogating cellular level metabolic models? So metabolic models are those models that are constructed to try to elucidate the metabolism of a cell. That's basically a subway map of all of the intercellular processes. Ideally, these can be created from annotated genomes where genes are converted to pathways and then are connected to elucidate metabolisms. So they're really complex and are really aimed at capturing complete biochemistry of a cell. There have been several published pipelines that allow for the automatic generation of these metabolic models. And for this study, we used the CarveMe software. We did a lot of method development on how to best leverage this tool. And unfortunately, I don't have time to talk about that today, but happy to take questions. So we use the CarveMe pipeline to build an ensemble of metabolic models for nearly 4,000 genomes from the Ocean Microbiomics database. And then we did our quality assessment, which resulted in about 1,600 genomes that we believe had generated high quality models that we kind of have confidence in at some level. And again, I'm happy to talk about this quality assessment, what worked and didn't work. It was a great learning process. And then what we did is we ran a large number of in silico tests with each of the models that we generated from each of the genomes in order to try to predict the metabolic strategy for each genome. So to do this, we asked the metabolic model, if you could grow on any substrate, what would you choose to grow on and how fast could you grow? So that's illustrated up here in the top right. And so this model had access to all different color compounds, and it decided to grow on red as its primary source with a little bit of blue and purple and the maximum growth rate of 1. And then we did these kind of knockout experiments, which we said, okay, well now if we take away certain compound classes, how do you respond? How does the model adjust? And what is the responding growth rate? So in this condition, we knocked out red and we ended up just with growth on blue and purple, but at a significantly reduced growth rate. So we did a whole suite of these tests and ended up with over 1 million, I put a $sign in there, I didn't mean that, over 1 million data points of kind of assessing the growth preferences for these different organisms. We then use an unsupervised learning approach, self-organizing maps or SOMs, to cluster metabolic strategies and try to identify common metabolic niches across this entire data set. And so these niches are based on substrate preferences. We also were able to assess growth rates based on codon usage bias estimated directly from the genomes. So before I get into the metabolic niches that we identified, I want to take a minute to talk about validation, which is obviously critical, because we really wanted to confirm that the models that we were generating, we could believe. These high quality models, we eliminated the ones we knew we couldn't believe, but we wanted to understand if these high quality models could reproduce experimental data. And so for this, we validated a whole series of models that we built for 146 strains that our collaborators, Manu Prakash and Otto Cordero, used in an experiment where they tested a large number of carbon substrates as sole carbon sources. So we had the experimental data of whether these strains could grow on these carbon sources, and then we ran our built models and ran them to see if we could mimic the experimental data. And we saw quite good agreement with a 76% perfect match of growth, non-growth between the data, the models and the experiments. We also looked at the growth rates predicted by the metabolic models compared to observed in the experimental data set. And again, we saw good agreement where for each strain, the substrates where the fastest predicted model growth rates also had the fastest observed model growth rates. All right, so to get into the niches that we identified through this study, we identified 8 metabolic niches, but for the sake of time, I'm just going to highlight a couple of examples. 3 here, a generalist, an acid specialist, and a sugar specialist. And what I'm showing on the left-hand side are these compound classes that we were testing. These are just 6 of the 11 that we tested in the study. And then these bubbles are showing the response of the removal of that compound class. So blue means that removing the compounds did not impact the growth of the organism as predicted by the metabolic model, whereas yellow showed a large impact such that without those substrates, the model predicted substantial growth reductions. So we identified this generalist group that had very flexible metabolisms. It seemed to be happy to grow on many different types of substrates and could kind of switch easily between different substrates, carbon substrates. This is in contrast to the acid specialists, where it was a clear niche that preferred to grow on acids, such that removing acids had a significant negative impact on the predicted model growth. And then finally, I've got this example of the sugar specialist niche that again really preferred to grow on carbohydrates, and the removal of that compound class resulted in significant growth rate changes. We can then take this one step further, and we can use that codon usage bias estimate to get at potential maximum growth rates for each of the genomes that fell into each of these niches. And we see that the generalists tend to be these fast-growing copiotrophic type organisms, whereas the slow-growing acid specialists in this niche tended to have much slower maximum growth rates. And then actually this third group had statistically significantly different growth rates that fell between these other two groups. So now we're finally getting to a place where I can envision putting this into a biogeochemical model. We've got a copiotroph oligotroph type of dichotomy, but with more insights into the metabolism and substrate preferences for these heterotrophs. For the microbiologists out there, we can also look at the taxonomy of the genomes associated with each niche. We see some expected trends. So the fast-growing group has a lot of classic copiotrophic organisms, like the Pseudomonalis, the Enterobacterialis, Flavos, and Rhodobacterialis, while the slow-growing group has, we see the Plagibacters, Marinosomotalis, and SAR86 showing up. What's interesting is that while we see some of these kind of expected trends, we don't see a simple phylogenetic separation, suggesting that, as others have before, the taxonomy alone is insufficient for determining function. So finally, we wanted to see in the real world, so these again were based off of this OMD database of genomes. We wanted to see how these metabolic niches translated into natural populations. And so to do this, we used the CalCoffee dataset in collaboration with Andy Allen's group, this long-standing repeat hydrography line off the coast of California. So to just orient you, this is what the samples that we're working with look like in terms of from the phytoplankton perspective. So this is phytoplankton community composition represented in an NMDS. And you can see the separation of chlorophyll along NMDS1, where the diatoms and high chlorophyll are here on the left, and then the prochlorococcus and the low chlorophyll samples are on the right. And then depth is separating out along the second axis, where we see high and low light probe separating out quite nicely. So we then looked at the heterotrophic communities associated with each of these samples and quantified the relative abundance of our 8 metabolic niches within each sample. And what we saw was some really, I think, exciting results. So we see that this fast-growing generalist cluster, the kind of copiotrophic cluster, was uniquely diatom associated, kind of in the bloom-forming regions, as one might expect. We saw that these colors are the relative abundance. I just kind of zoomed in on this little node here of this group. So the slow-growing acid specialist associated with the low light adapted pro. And one of the other niches, which I didn't talk about in the previous slide, is another slow-growing specialist. It has some vitamin oxytrophies and other unique substrate preferences, uniquely associated with the high light adapted pro, and is a distinct group from that associating with the low light adapted pro. In fact, we could account for about 85% of the heterotrophic community using our 8 metabolic niches, which is suggesting that these niches might be a good building block for understanding the microbial communities in situ. And so as we move up in scale, the next step is to really try to take these metabolic niches or metabolic functional guilds and put them into a global biogeochemical model. So this is work that was led by my former postdoc, Emily Zakem, who's current faculty at Carnegie, and was published a few weeks ago in Science. Sorry, I'm not high-tech enough to put up the QR code, but you can find it. So this was done actually in parallel to the studies I was just telling you about. So we used a slightly different definition of metabolic niches or functional guilds for the study. And that was driven in large part because we're still trying to figure out how to best convert between model DOM and chemical compounds that we measure in situ or think about experimentally. So in Emily's study, we used 5 different compound classes based on their livability. And then we had 2 different types of heterotrophic functional guilds for each, a gleaner or kind of oligotroph, and then a opportunistic or copiotroph for each. So we had 10 different heterotrophic functional gills that were then included in the Darwin ecosystem model. So we can then compare the global, and this was run for global prediction. So we can compare the global predictions of distribution for these different functional groups to what we can see in observations from the distributions of ASVs. So what I'm showing here on the top panel is the model DOM, so predicted model DOM concentrations, and then observed on the right. And then just 3 of those 10 different functional guilds in the model compared to the corresponding ASV clade that showed the similar pattern. And so you can see that we see very different, as we would expect, patterns in the ASV data that's actually mirrored in the model. But now we have underlying dynamics of what is driving these rates of growth and consumption of organic matter and substrate preference for these different groups. So I'm really excited about the combined results of these studies. I feel like we are finally able to free up the kind of intellectual logjam that we've had that's long prevented us from being able to think about how we put heterotrophic microbes into biogeochemical models and how we incorporate the amazing omic data that's coming out of the oceans and help inform a large-scale biogeochemical model. So in the last few minutes, I wanted to tell you briefly about some other work we've been doing with a different group of cellular scale models that really allow us to dive in and understand the trade-offs that are associated with different metabolic strategies. And so for this, we've been using proteome allocation models. So these are really just optimization problems. I find them super fun and cool to work with. So unlike the metabolic models I just told you about, the proteome models have a much more simplified metabolism. So they are more complex than the global biogeochemical models or even the ones that have intercellular components. And are aimed at representing the core processes that are required for growth. So substrate uptake, biomass synthesis, energy demand, et cetera. What these models are really good at is providing hypotheses as to the optimal strategy for growth given an external environment and identifying the associated trade-offs. And so you can actually put any kind of metabolism that you're looking to kind of understand the trade-offs between into these models and investigate and generate hypotheses. So the model solves for the optimal metabolism and associated growth rate and cell size. So for example, if we have a cell here that I just drew out that can use either substrate A or substrate B, we can ask how it will grow optimally in environment 1, where maybe it will prefer to use substrate A, have small cell sizes and higher growth rates, versus environment 2, where it might switch to using substrate B, have larger cell sizes and lower growth rates. So we've been using these type of models to investigate a bunch of different questions in the group. And what I wanted to tell you about is some work that my postdoc, Susanna Lelas, who's in the process of moving to UT Austin to start a faculty position, has been doing using a proteome allocation model of phytoplankton to try to understand the trade-offs associated with multi-stressor growth. And we propose that there's a trade-off that cells have to struggle with, specifically under nutrient limitation. I think that we all might kind of intuit that cells need to get smaller to maximize their surface area to volume and maximize uptake of a finite resource. In contrast, under heat stress, we believe that cells have a selective pressure to get larger in order to minimize damage and have space to have the processes that can help mitigate ROS damage, for example, and also store more carbon resources for respiration demands that are elevated under heat stress. And so this becomes a challenge for cells. They can't do both at the same time. You can't be both small and large simultaneously. So for this model, we have our phototrophic cells that they are required, or they acquire light, photon energy. They fix carbon, and then they also have to take up nutrients from the environment in order to build biomass. Each of these blue boxes is a protein pool, and each arrow are the fluxes that are optimized through the optimization. What I think is also really exciting about proteome allocation models is they can directly validate against both physiology data. So as shown here, the lines are model output, that all of the symbols are observations of a bunch of different types from growth rate, chlorophyll content, and to C ratio. But we can also directly compare against omics data. So this is a table comparing how the model does to predicted shifts in nutrient limitation compared to the observed proteomic observations of experimental data. And we can also do similar comparisons to transcriptional responses and see good results. So for this study, we used 3 different phytoplankton types. We have a kind of nominal cyanobacteria, and then a warm and cold adapted diatom. So to generate some hypotheses on how cells might manage this metabolic trade-off under multi-stressor growth, we solved the proteome model under present and future projections of temperature and nitrate concentrations from the CESM model across the latitudinal gradient and the Atlantic. And so the black lines here are the temperature under present day with turn of the century, and then end of the century in dashed, and then nitrate on the right. And so here's an example of some of the model output that we get when we run the proteome model along these gradients. These are the optimal growth rates for each of the modeled phenotypes, and we also have the underlying predicted metabolisms. But in the interest of time, I just wanted to focus on 2 key findings. The first is that all the phenotypes are predicted to get smaller under future conditions in the temperate and polar seas and in the higher latitude regions, where nitrogen stress is predicted to be more severe than heat stress. And this results in the poleward expansion of the cyanobacterial phenotype as been predicted by other models, modeling studies. However, we also predict that all phenotypes will get larger in the kind of equatorial regions where heat stress becomes really severe and a larger challenge to deal with than the nutrient stress. The model suggests that the cyanobacteria, predominantly Prochlorococcus in the tropics, are more vulnerable to the heat stress due to their low heat mitigation capacity and small cell sizes. And so we identify potentially a new niche in these warm tropical seas where larger phytoplankton, Synechococcus, or even small eukaryotes might have a competitive advantage due to their larger flexibility in cell size, thermal tolerance, and their ability to mitigate oxidative stress. So this scaling of cellular decision-making to global biogeography has allowed us to present different hypotheses about how phytoplankton distributions might change and suggest that we might want to go back to biogeochemical, these large-scale biogeochemical models and explicitly incorporate the impact of heat stress so we can kind of dig into this a bit further. So I know this has been a bit of a whirlwind of information, but I hope it's gotten you thinking a little bit about how we might use models to bridge these scales from the cellular level up to the global scale. This is a figure from a recent Annual Reviews article that I think nicely illustrates how we can leverage many different types of models in conjunction with many different types of observations and experimental data in order to complete this picture of biogeochemical cycling that we really need to figure out. And so hopefully through this kind of integration, we'll get a better handle on these purple arrows and better be able to predict the kind of carbon fluxes that we need to understand. And so with that, I want to thank my amazing group who has done this work and collaborators, our funding sources, particularly the Simons Foundation and NSF. And with that, I'd be happy to take questions if we have time.
Thanks, Naomi. That was awesome. And you're under time by a minute or two, so we do have time for maybe one question. I don't know if anyone wants to raise their hand and then speak up. There's questions coming in on the chat as well.
Can't find the chat window right now, sorry.
Let's see.
I've lost the chat. Oh, here we go.
No one's gonna raise their hand? I'm amazed by that. Maybe it's just we've been rushing through so fast because next we have the panel discussion anyway. So
The Psalms are not topical.
does anyone like Maria or Jim, do you want to come off mute and ask your question? Or can you see him?
Yeah, I found it. I found the chat.
Sorry, I couldn't find my hand to raise. Okay. You read the question though.
Yeah, no, it's such a good question. Yeah, so we did a lot of work with the Psalms and other clustering techniques. And yeah, you're totally right. You have to be really careful with them. The short answer is I think it's robust. It actually shows the same patterns as other things like PCA and hierarchical clustering. The Psalms is nice because the data actually, it just so many data points, it handles it a lot better than the other methods, but it's not, we actually don't see much nonlinearity in it. We see, in general, the models are either insensitive or quite sensitive, and so it's almost a binary data set. It's not really, but like there's not a lot of instances where the metabolic model predicts that if you are reducing the substrate concentration, that it uses a little bit of it and then also switches using a little bit of something else. It's either like, I want to use this compound class, or I'm going to switch using something else. And so I think hopefully that answers your question.
There are more questions coming in, but I'm going to suggest.
Yeah, I have a quick question.
Go ahead, Jim. Yeah.
And that's really the question about haptophytes or coccolithophores. I noticed that you talked about diatoms. I was wondering what do you think the fate of coccolithophores is?
Yeah, so I think that yeah, I guess I should have clarified. We have a lot more data in order to validate the diatom proteome model. In particular, we wanted to make sure we could validate against these proteomic data sets, which we need more of because we didn't have many. So I think I would hypothesize the coccolithophores will act similar to that kind of diatom class. I guess the main takeaway I'm hoping you'll get is that I think Pro is amazing in current oligotrophic oceans, but I am not convinced that the niche that Pro is very good at is going to be the same niche that we're going to see in the future tropical oceans, and that other organisms might have a better strategy for dealing with that multi-stressor scenarios.
Cool, thank you.
Okay, thank you so much, Naomi. I'm gonna hand this over to John Lauderdale, and I think he's gonna open up for a panel discussion so we can keep going on the questions for Naomi and others.
All right, thanks, Dan. Yeah, if we can have our speakers turn your cameras on if you're able, and we can field some discussion across all sorts of scales. All right, so I guess as usual with OCB, I've come away completely blown away by all the different aspects of what we've heard today. So given that there are plenty of what we might consider known unknowns, as in trying to link all the small scale stuff to the large scale stuff and making progress through observations and models, what do the panelists think that unknown unknowns might be in bridging scales across the ocean carbon cycle? Does anyone want to take a first go at that?
I can say something. Is it just you and me, Melissa, I guess? I think, I mean, yeah, again, have been really blown away with the observational, like Melissa's talk and Manu's talk. I think that is like just skyrocketing us forward in our ability to visualize and see what's happening. And I think that's going to be critical. I think I don't know if it's an unknown, but from my perspective, the lack of information, so we can now go out and sequence lots of things, right? We can understand kind of who's there, but then if we want to actually understand what they're doing, so we can link it to these images and other rates that we can measure, we are only as good as our annotation ability. And so I think that, in my opinion, is like one of the key things that is an unknown. We don't actually know what they're doing. We're basing a lot on E. coli and kind of these handful of model organisms, which don't apply. So what I didn't show you with the kind of quality assessment is that the models, the automatically generated metabolic models, do really well for things that look like E. coli. And they do really crap at things that don't look like E. coli. And we have a lot of things in the ocean that don't look like E. coli. And so I think that's kind of where I think the biggest like limitation is personally.
Yeah, I would just add to that. I mean, it's hard to know what an unknown, you know, because the people about it. But my feeling is that it's is that we have these tools, and one of our biggest challenges, just like in the past, has been to like link these things together. So link the biogeochemistry directly to, and the sort of functionality of the cell directly to the images that we're getting. So we get all these little like pieces, and we build stories out of them and try to make sense out of it. And I think that work will just continue with some of the new capabilities that we have, modeling and observational.
I was going to continue the question about unknown unknowns, and I think the most clear place where we don't know anything is breakpoints, because we're trying to predict, and there are just an entire industry of people trying to predict how things will change. But breakpoints or change points is something we just don't know where it's going to happen. So that is the biggest problem. And that is also the most frustrating, because that's the hardest thing to understand. What's happened if we have a 5-degree warmer ocean? We don't, no one knows. It's impossible to know, and especially with nonlinearities.
So Maria asks, do we use machine learning inefficiently, and where we use it only to optimize prediction versus using it in novelty detection or to generate hypotheses? And I would add another one that follows on from Dreux is, can we use it to kind of, is it difficult to be able to believe what comes out of machine learning based on, out of sample type events like a huge warming, a heat wave, or a MCDR type application?
I guess I'm going to take kind of a middle answer. As a kind of mechanistic modeler, I am very nervous for when we go machine learning into like future worlds where we're unconstrained. However, I also think that we are inefficiently using machine learning, and we're not using it enough to generate hypotheses. And I think we as a field maybe tend to err on, if we know that they're not perfect and it's really hard to do some of these problems, that we kind of throw the baby out with the bathwater, opposed to trying to understand what we can get out of some of these automated pipelines, and how we can leverage what we do believe and discard what we are skeptical of. And so I think I'd love to see more of that.
Is Hongjie online? Can she maybe chime in? Okay. Jessica, would you like to ask your question?
Thanks. Great presentations, everyone. If Mara Freelick is still here, there was a little chat in the morning following Elizabeth Yankovsky's talk about scale-dependent parameterizations for ocean biogeochemical models, and I would actually like to pose this question now to the panel, including Naomi and Mara. What do you think about this? We don't really include any scale-dependent parameterizations in ocean BGC models. Maybe kind of historically, these models were resolved on very coarse scales, so these small-scale interactions are not included. Like based on your work, could you comment on this a little bit? Like, should we include it? Like, should we be moving towards including these small-scale dependent parameterizations?
All right, I can jump in. I missed the very end of your question because we're actually in a group room, so I was switching audio. But I think I got it. Just asking, should we be including these scale-dependent parameterizations? Yeah, so I think what we're missing in biogeochemical modeling, fundamentally, it's about the interaction or covariance between tracers. So if you're talking about nutrient phytoplankton covariances, or particularly the work that we've been doing is vertical velocity and nutrient covariances. All of those are scale dependent, and you miss those if you are just doing a parameterization that's diffusive rather than accounting for the reactions that may be happening at those small scales where you have a covariance, say, of vertical velocity and nutrients leading to productivity or nutrients, phytoplankton, zooplankton. There are ways that we can account for those. And so we've shown in some of my recent work and work with a student that gives you a dependence on growth rate that's unaccounted for and creates sort of an emergent nonlinearity. We can account for those, and we've been developing asymptotic models where you can account for those covariances using the information you have at the large scale. Obviously, you can't get everything, but you can, very similar to the way that we've been doing physical parameterizations, account for reactions. And we're not making use of all the information that we could be using. And so we've been working on those mathematical approaches where we could be making use at least of all the information that we could be using for biogeochemical models, and doing that in a general framework. So hopefully that made sense.
Yeah, I think Mara, I think your answer is great. Yeah, I think the key thing is trying to figure out where the nonlinearities are and which processes generate nonlinearities, I guess, kind of to Boris' point a little bit ago. And those are the ones we have to figure out how to incorporate a scale-dependent parameterization that's going to capture those nonlinear responses, opposed to just kind of more of a linear process that can be parameterized in the kind of more conventional ways.
Thanks so much. There's lots of work for all of us to do.
I think Elizabeth mentioned about using the Oceananagons model that's very efficient and can run really fast on GPUs. So is this scale-dependent case literally just adding more resolution until you capture it? Or, yeah, how can we determine how much is enough resolution? Might we also need resolution in sort of ecosystems or functional groups or something like that as well? To what resolution is enough and what can we parameterize?
I see Boris' hand up for a little while, so
Yeah.
I don't know if that's...
Sorry, I was just going to do a little bit of backtrack to machine learning. I hate to do these things, but I think it's really important to emphasize that when we work with machine learning, many machine learning technologies, they don't extrapolate. They only interpolate. And you can do this. It's a very simple thing you can do. Like if you take a random forest regression, for example, and you have you have an independent variable, and then you predict the dependent variables from that, you'll see that as soon as you get out of the dimension space of the independent variable, the model starts to flatline. And that is something that's very good to show to students, because that means that you can never use these methods explicitly, but also other methods implicitly to see a new state. If you haven't experienced part of a multidimensional space, you don't know what's happening for the machine learning method. And I've been in meetings where people have presented XG boosting methods to predict extreme wind events, for example. And they get really, really good results everywhere. But you're like, yeah, but you don't know, you can't get a stronger hurricane from your data. It's not possible with your method. So that is something to really, really think about. It's like, where does whatever model you're using, where does it break?
Yeah, totally. I'm from not a machine learning person. That makes me very nervous. And I think there's a couple of comments. Jan Giacomo Navarro says, I think machine learning can be a great tool to add variability in parameterizations or generating new parameterizations, which is a great point. But as for every other tool, it can't be used blindly, and it's important to emphasize explainability. Yeah, so I think I see Hongjie and Elizabeth are online. Would you guys like to chime in on the machine learning and what processes we think we need to target with those approaches?
Yeah, I'd like to, oh, Hongjie, you can go ahead, sorry.
Sure. Thank you. Sorry, I walked my dog a few minutes ago. She needed to pee, and I heard some like my name was called. Anyway, so I think I'm not quite familiar with the machine learning too, but I always worry about the data set to train the machine learning. Like, for example, the data, the map I showed this morning, like there's the data limitations, like it's big limited data limitation. And if you only have, for example, a 3-month data set, how could we trust the prediction based on only 3 months data set to to present that represent the true natural variability? But that said, that's the prediction based on the best understanding. So it's a tool, we can use them, but I really think we need to use with caution. That's what I would want to add.
Yeah, I want to second that as well. So I've worked a lot on turbulence parameterizations, both for submesoscale and mesoscale processes, and have read a lot of papers and seen kind of efforts where scientists try to apply machine learning to really parameterize subgrid turbulence. And I very much have kind of seen that a lot of these machine learning parameterizations end up having to impose physical constraints, such as energy conservation or some sort of entropy, I don't know, laws. And they end up really just mimicking existing physics parameterizations that we already have, and not really teaching us anything new. So I think that for turbulence parameterization, especially for turbulence that varies pretty significantly across scales, I have not yet seen any convincing application of machine learning, and I really believe in physics-driven approaches there. But I do think kind of as I alluded to in my talk earlier, I think that the forecasting aspect of machine learning is pretty interesting. And the fact that when you're doing very short-term forecasts where you're not taking the machine learning algorithm out of the type of data or the type of environment that it was trained in, there it can be pretty powerful. And so I think using things like downscaling to get higher resolution fields from existing climate model simulations that we know actually capture the physics properly can be a pretty compelling use case of machine learning. But I do agree that it's, in general, I haven't seen it discover new physics or be able to really have any fidelity in longer term climate predictions.
I know another application of machine learning that, so Galen McKinley says that machine learning have to be careful. It's a tool and you have to understand your tool to use it appropriately. So one crucial aspect of machine learning is being able to fill in sparse data gaps. And so for the MCDR type applications, one way that I can see it being used is to help you sort of fill in the gaps of sparse PCO2 measurements to try and do some MRV type exercises. Are we comfortable with that, or does that ring alarm bells?
Yeah, I mean, I think we had a lot of success using sparse SOCAT data and interpolating it to global scale with machine learning of various types. But it doesn't capture all the timescales. It captures the mean pretty well, it captures seasonality, but it doesn't capture the decadal variability as well as we would like. And so then we need to think about where we need more data. There's really a variety of ways we can use machine learning, like parameterization, equation discovery is one way. Parameter estimation is another really promising way that they're making a lot of promising progress on, for example, with CSM right now. And then there's also, of course, the interpolation. But we have to think of ways to ground truth what we're doing and understand the approaches. And just because an XGBoost doesn't extrapolate well, doesn't mean that a neural network doesn't extrapolate well, right? And it might, for certain situations, do a pretty good job of generalization to some point. But we have to figure out how to test it, right? So I think that, it's just like saying, I think the question of isn't machine learning a good tool? It's like asking, is linear regression a good tool? Yeah, there are things that linear regression works well for, and there's things that you wouldn't want to use it for. And we, as our community, I think, needs to become more sophisticated in our use of it and understanding of it. There's 1,000 different ways. And we really shouldn't just say machine learning. We should be talking about machine learning for this or that or something else. And there's not one thing. It's so many different things. It's not just parameterization. It's not just equation discovery. It's not just any one thing. So I think we really need to educate our students about machine learning and turn it into a tool that we can use for many different things.
So Melissa, do you want to say what you were thinking?
I would just put in the chat, but I mean, this is just about the fact that, a lot of the machine learning methods and tools require a lot of compute power. And I just wanted to highlight that there are sort of some methods for introducing machine learning onto little microcontroller chips as opposed to computers or other things that require GPU. So I think that that's an exciting area that we might see emerging over the next little while, and it would enable, for example, even sort of lighter weight in a sense platform than the current Minions and SnowCam that I showed today, where you could have much lower power. I mean, probably by a factor of 100 and still achieve a lot of the things that these things were doing.
Yeah, that's fantastic. And then you could generate even more observations. So that would be really useful. Sorry, we didn't catch that, Melissa.
I just said yes.
Oh, okay. I think you're not sure if you were muted. Okay. Bro, you have a question or comment?
Well, or a comment, yeah, just to continue on what Gail was saying, that one thing that is becoming more and more clear the more you work with new statistical methods is that it is a signal to noise ratio question, or like the question about how much of the variance that you can describe with the data you have as input features. For example, we've been working a lot with both PI parameters and primary production estimates and using machine learning or other ways to get better satellite primary production models. But you don't have nutrients. You don't have nutrients. There's no magic in it. You can't just like create get around the fact that we don't have a way of getting to nutrients from satellites. So that means that we need to figure out, one, why is the satellite models working as well as they do? Why can they do that without knowing the nutrient state or the nutrient limitations at a given time? And two, what does that mean as a limitation, and as a limitation in dimensional space as well? And we've had some really harsh conversations about that in some groups that I belong to. But it is a very interesting question, and it's very close to what we're talking about when it comes to machine learning. But it's also a broader question about what is it? What kind of data, what kind of information do we need to better understand the systems we're interested in? I always get back to mixed layer depth dynamics on short timescales, because if we had that, we could do so much better in so many ways, but we don't, and there's no way to get around it. We don't have it, which is super annoying. Can someone get that for us?
What would you need? Like a large observing network of expensive floats, or maybe
So exactly. LiDAR on floating devices.
I'm not sure how much
Okay.
the speakers are supposed to be talking. I feel like I dominated so much of everybody's time already today. But Suzanne, if you want to
go for it.
Yeah. Hey, hi there. Is it okay to just jump in? I wasn't sure if you could see my hands or not.
Yeah, go for it.
So I'm finding this conversation about machine learning really interesting. I use it a ton, but I'll tell you, full disclosure, I am not a data scientist. I'm not a computer scientist. What I do is I collaborate in a 50-50 collaboration with my computer scientist and data scientist friends and colleagues, because just as Galen said in the chat, you have to know how to use your tool. I'm a classically trained physicist that kind of morphed and reinvented myself into an oceanographer of a biological bent. But I think until, over the next couple of decades, until we come out of tertiary learning institutes, universities, skilled in both machine learning and our domain speciality, that's not going to happen for a pretty long time. Until then, we have to be leveraging the skill and the power of our computer scientist friends and colleagues. Something that we're doing a lot of in our lab right now is we're taking on computer science folks that have got backgrounds in image processing, and they are bringing completely new tools to bear on very old problems that we've all struggled with, in the various aspects of oceanography that we study. So I would say, and until we come out with, until we all graduate with double majors in data science and our domain speciality that leverage the power of the data science communities. They are a really fun bunch of folks to work with. We're the domain specialists. We can tell them what is realistic in terms of ecology, biology, physics, chemistry. But they really know how to leverage the tools that are rapidly evolving in this space. So that's just a comment on from my point of view.
Okay, Hongjie.
So I just want to make this Suzanne's comment more colorful. Last time I talked with one of my friends from computer science, and I complained to him like our computer, like our model simulation is so slow. I need to wait 1 week to get until I got a very, very reliable simulation. And he asked me, 1 week? We only tolerate 10 seconds searching time span. So if it's longer than 10 seconds, that fill our requirement of like 10 seconds versus 1 week. And kind of I think we're talking different things. And after I talk with them about the way we do the simulation, coastal ROM simulation, like there's definitely way to improve to make it more efficient. So I just want to lay this this kind of a conversation here. So hopefully people can pick this thread, and maybe they will develop a new way to run models. For them, 10 seconds. That's something like a Google search. Like if you wait more than 10 seconds, you start on the engine, you try a new search. But sometimes I want to add here.
Yeah, that is a great point. I've had similar conversations where my data science and computer science colleagues, they quietly and politely kind of go, oh, guffaw at me and then tell me how it should be done. So no, absolutely, I've had a similar conversation.
And I think that's Galen's comment about in the chat about where's it gone? Yeah, doing engaging people who want to do problems in climate science and stuff like that to help advance us in that respect. Yeah. Mara, go ahead.
Yeah, I guess I have a question for speakers, both Naomi and Melissa, and it's related to this machine learning problem where I think we're often in a case of data sparsity, even with this image data and with genomics data. And so there's a lot we can do with that. But I was just curious of hearing your perspectives on the sorts of approaches, both in modeling and observations, to deal with the data sparsity where we're not quite reaching the statistical power that we would like to have.
Yeah, I think it's a great question. I mean, I want to come back to like Maria's original question, which I think was a little bit, we've sort of ranged a little bit now in terms of a general discussion on machine learning and how good is it? But you know, I think that her original question was about how to use machine learning in a way to generate hypotheses rather than just sort of use it for parameterization, or sort of the notion of using it to fill in gaps and make predictions in areas that, you know, might have a sparsity of information. I don't know. I guess I'm like a little bit more hesitant in that way because I feel like I have so many. I mean, what I would love to do with the tools that I have is just like every time we have the chance to look more closely at something, right? It's like Manu's talk was a great example. Or if we improve our resolution on our cameras or do something where you see this technological improvement that then reveals new information. To me, that is so incredibly exciting. And I'm not sure that I can envision in a setting where I would be able to just apply a machine learning tool to that type of problem and have it reveal something completely new, or at least I would have trouble trusting that. And so I use these tools in a maybe in a less creative way in terms of just trying to use it for image identification, like ROE identification, classification, and just to really expedite the computational demands of image processing. And there's so much to be gained just by developing those tools. Like we will learn so much. Right now, we have such a excess of image data that is sort of sitting kind of unprocessed because we don't quite yet have all of the tools needed to be able to understand and make sense of that. So I think there's a lot that can be done just in that realm.
I totally agree. And Mara, I think you asking is like such an important question. I'm like really waiting for, I feel like someone out there, you must be working on using these kind of techniques to help us figure out where we are missing vital information, when we think about sparse data, right? So that seems to me like a really useful application, right? Under present day, we know what we know in the places we've gone, and we can use some of these techniques to generate hypotheses about where we may need to sample to better constrain the system that we want to constrain. And so is extrapolating outside the bounds of what we've sampled, but within our current Earth system where we think we understand kind of how things are functioning. And I think that that's an ideal example of using these processes to generate hypotheses, right? And then you can go out and check, and if it did a good job, then great. And if it did not a good job, then we know we need to dive in further.
Thanks. So there's a bit of conversation in the chat about using machine learning in an exploratory sense, which we should be exploiting much more. I think I liked, Naomi, you had a section where you used kind of machine learning to generate different models from genome data, and your comment about functionality doesn't necessarily predict who is there. I wondered if you could expand on that.
Oh yeah, that was just that. And again, we're not the first to have shown this, but our metabolic kind of functional guilds that we predict have some taxonomic links that we would expect, but it's not that you can't go straight from taxonomy to metabolic function. You need to kind of understand, there are multiple groups within very different taxonomic groups, or clades within different taxonomic groups that could be acting similarly in your system. So that was that point.
So does the reverse happen as well? I guess that's the question. So if you had, for example, lots of taxonomic data, that also does not necessarily lead to functional.
Yeah,
so
I mean,
that's an AI kind of thing
yeah,
that
I
we need
mean,
to be careful about.
That is a really key question that I think is, in my opinion, a really active area of research, is how we can use metabarcoding or ASVs, and the data is very easier to generate than some of these other measurements, but maybe doesn't have as much information, and how we can leverage that. And maybe again, that's a way that AI could be really valuable, to say, okay, this is a kind of lower cost, higher throughput measurement that doesn't give us a complete picture, but tells us something about the community that's there, and then how can we expand upon that to get the actual biogeochemical picture that we need.
Cool. So we've got a few minutes left. I just wondered if the speakers could give us some sort of perspective on the future of research bridging time and space scales. So I don't want to put anyone on the spot, but we go in speaker order. Hongjie, would you be able to say something about future perspectives?
Okay, I can try first. So I do think no matter what a CDR approach we want to target in the future, as long as scale up to gigaton time scale, that's a huge perturbation for the nature environment. So we should always keep the bigger picture in the mind, like how this, for example, like a nutrient, like either isolation or carp cultivation. So as long as you touch the biology, the ultimate bottleneck is the nutrient variability, availability. So yes, we can speed up the algae cultivation, for example, but how will that impact the phytoplankton? I think as long as the CDR scale up to gigaton time scale, all the interaction, the big spatial or temporal scale of issues should be something we should keep in the mind. And so that's something I would like to throw the thread here. So the big picture should be always behind this MCDR, and because we are use as MCDR to scale up CO2 removal on gigaton time scale.
Okay.
Yeah.
All right, thanks. Elizabeth?
Yeah, I guess something that I wanted to bring up again is just I find that there's an interesting tension right now between some of these traditional kind of modeling efforts that have been long developed in modeling centers, and how now a lot of these efforts are getting shifted partially into industry, partially into startups, or kind of various organizations trying to develop their own AI or modeling infrastructures. And I find this kind of interesting and really wonder about how the future of climate modeling and kind of climate prediction will evolve, whether it will continue to be developed in modeling centers and academic institutions or really move more into the private sector. So I think that's an interesting question, but I do think fundamentally it's really important to maintain process understanding and not allow some of these efforts to move out of the science scope, and to have scientists continue to work on them and to do idealized models and process studies and really deeply understand the dynamics, because I do worry if we focus too much on AI and some of these flashy new modeling techniques that we will lose a lot of generational knowledge that has been developed over many decades.
Cool, thank you.
And
Is Kristen online? Sorry.
That's okay.
I don't think. All right, Melissa, go for it.
I was just eager to follow along
Yeah, right, exactly.
what Elizabeth said, because I think the same is true for our technology capabilities, our observational and in situ measurements. I just think that it's so crucial that we don't, that we maintain, a strong presence in terms of making direct measurements of the ocean.
And last but not least, Naomi.
Yeah, I love all of those thoughts. Yeah, I think that I've been excited about the kind of groundswell of like truly interdisciplinary studies that have been happening over the past quite a few years now. And so I think continuing to like bring the fields together and making measurements simultaneously. Well, also I think, Melissa, I felt like your talk was really inspiring, that we need to make sure that we're developing low-cost solutions so that we aren't kind of pricing out studies from being able to make some of these measurements. If there's only like one group in the entire world that can have this super cool technology, that's not going to allow us to sample the ocean. And kind of also to Mara's point, in the way that we need to sample it to understand and get to that mechanistic level, we need a lot more samples to be able to kind of dive in and do those predictions.
All right, thank you very much, everyone. Round of applause for all our contributors to the discussion. Fantastic.